Well, thank you so much, Angus. Here we are on day four, and it's been just an amazing journey, I think, for all of us. Um, I've just found it so invigorating myself. I hope you feel the same way. One of the things I was talking to others about yesterday is I just really appreciate the sort of emotional support you guys are all giving each other, hearts flying around and clapping hands and such after people are presenting. And I think that's so important because for many folks, this is a real culmination to be able to show their work, tireless uh, long work into long hours, hard days in the field, many days analyzing in front of a computer, all that just to be able to show and share it with you. So I think that's amazing. I also love the international interaction as we have, questions coming from Scotland to South Africa or from Brazil to someone in Indonesia, for instance. And so one of the things that I'm thoughtful about as we're on our last day here is, how do we stay connected? And one of the thoughts that I have is that many of you will be connected with folks in your own country or working on the same species, but maybe with the same theme, whether that's ecotoxicology, community engagement, habitat loss, genomics, you name it. Maybe there are certain groups that can form, connect, I don't know how, WhatsApp or other fora, so that you can look at the same kinds of challenges really across different landscapes and different situations. I should have said, and I'm getting too comfortable. I'm Axel Murray-Ramcharan. I'm director of the Small Cats program and conservation translocations for Panthera, just for any of you who are just joining today. And it's been such a privilege to have all of you and many others that have joined over the other days with us. Today we have an amazing lineup again in terms of a keynote that I'm very much looking forward to, then short and longer talks, of course, and then a panel, this time looking from detections to density and looking at how we rethink how we measure small cat recovery. So here we are. We're ready to kick off. And no better way to kick off than with our amazing keynote today. I'm so pleased to have with us Tadeu, who's a professor of the Wildlife Conservation and Ecology Lab at Marina State University, researcher for Instituto Pro Carnívoros, Pro Vida Brasil, and member of the IUCN SSC Cat Specialist Group and Canid Specialist Group. He's a founder and chair of the Tiger Cats Conservation Initiative, which focuses on the conservation of 9 small felids in the Americas. His interests include the ecology, conservation, and natural history of neotropical felids, carnivores, and endangered species. He leads a long-term, multidisciplinary, continent-wide conservation program focusing on ecology, biogeography, natural history, and conservation of the 9 small felids of the tropical Americas. Of course, my Portuguese is terrible, or non-existent, but Projeto, Gatos, Domato, Brasil, Americas. I see Tadeu laughing. You can correct me and give me some language lessons sometime, which contributes to their conservation, status assessment, population monitoring, and understanding of their ecology. Thank you so much for joining us.
Okay.
Great, thank you. Perfect. Looks wonderful.
Okay. So we're going to be talking about conservation action and conservation science, aiming at small cats protection, and I'm going to use the Tiger Cats Conservation Initiative as a model. The question is, do conservation action and science mutually exclude one another? That is the main question for researchers, especially in academia. How academia sees and handles conservation actions. This is something that I think has been missing. And when I teach conservation biology, I always tell my students, listen, if you do research, even if it's focused on conservation, but you do not do conservation action, you're missing something. And I think academia has to wake up some more for this. And what I'm going to show here is what we've been doing that I believe can accomplish this. In the Tiger Cat Conservation Initiative, we have several targets to ensure long-term conservation of the population, monitor population, to have partnership with landowners, because a lot of small cat protection is done outside protected areas. So it is highly important to have them by our side. The environmental education program that we have, they will see some of it, habitat monitoring, population monitoring, looking for population trends to see how the cats are faring, to establish a permanent neutering and vaccination campaign, our One Health approach to conservation to focus on the multiple aspects, and training of professional, the students, community, little kids, everyone interested in community conservation. Our actions are built upon action and vigilance. Action in conservation action taking to reduce threats. So we have the threats, and vigilance to monitor key population of the threatened. The focus is on the tiger cat species complex, but it also includes several other species, as I will show you shortly. So we have the threats, where you can see in slide, and we have the action and vigilance. In that slide, we have both action and vigilance, because you see a little kid, which is part of the community, and his father that also is part of the community that works with us. So at the same time that we have like conservation action, because this is one of the part of it, this is the monitoring part. So I chose this slide because it depicts both. Here we have the other species we cover. As I said, Tiger Cat Conservation Initiative could also mean Tropical Cat Conservation Initiative. As I said, the focus is on Atlantic Forest Tiger Cat, Clouded Tiger Cat, Savannah Tiger Cat, but also includes Margay, Ocelot, Grundy, the Cerrado Black-Footed Cat, which is Bracatus, the Pampa Cat, Munoai from the Colocola complex, and Jeffrey's cat in a very small scale. The environments the tiger cat complex lives and where we target most of our actions are pretty much endangered by themselves. And for each tiger cat species, we have our strategy is to have long-term conservation safe haven. And for the clouded tiger cat, we have La Amistad, the La Amistad complex in Costa Rica, Panama. And for the Costa Rican Oncilla subspecies, for the mainland in the northern Andes, the three cordilleras in Colombia, which is another key area in each one of them. For the Atlantic Forest tiger cat, that would be Caparaó National Park area and its surroundings, Aparados da Serra and Serra Gaúcha Mosaic. And for the Savannah tiger cat, that would be Mirador State Park, which where I am now, and Capivara Confusões National Park Complex. So with these, we aim, let's say, Mirador can withstand a self-sustaining population. But first, let's see conservation in action that we've been doing. Doses of vaccines, animals vaccinated, and animals neutered. These are just estimates. Animals vaccinated are pretty accurate. Doses of vaccine are approximate because it depends on the region and how many doses are applied. Animal neutering, we are expanding our neutering now. We have our own neutering clinic, mobile clinic, that we built with the grants from the Mohammed bin Zayed Species Conservation Fund. That's why we named it the Mohammed bin Zayed mobile vet clinic. These are some actions for our several campaigns. We also do, with our vaccination campaigns, we also do the intercontinental vaccinations. The last one we had projects from Central Mexico to Central Chile. And not only within the Tiger Cat Conservation Initiative projects, we also have participation from other working groups, from several other projects, from several other working groups. And we will keep doing these. What is the purpose of this? When you do vaccination by yourself in your project, it's one thing. But everybody, at the day we did the first time, they said you have a sense of something else, that you did something special. It's one thing when you're doing at your study site, but it's different when you're doing at your study site connected to several other people all over the continent. Actually, covered the three Americas: North America, Middle America, and South America, because Mexico is in North America. And it gives you a powerful sense. And with these campaigns, we start showing authorities. And now we are partnering with counties for vaccinations. We are always being asked to start doing this. And we focus the vaccinations around the key areas we want to protect, because dogs are pretty much everywhere. For our environmental education, we focus on, especially on children, because we say that children are our future. And we develop a series of games and other material using the principle that learning could be fun. And this has become very popular. They love to dress up, to put the mask of the tiger cats. And we have not only of the tiger cat, but of all other species of small cats in the areas. The numbers you see here for people reached, it includes not only 1 year. The other numbers are only for last years. But these, especially coming from the huge program we have at Felinos do Aguaí, that has been going on for several years. That's why we have like 42,000 people reached. But it's very popular. It's everywhere. In most, I would say, every project has an environmental component, environmental education component. We have also a series of workshops and participation in varying events, from COP to classes to community, from workshops related to communities, workshop at academic level in many ways. And also here, there was a meeting of the working groups from Latin America, from the Americas, where we had the chance to meet some of our donors. We got to meet Ming from Panthera, that has supported our work, and the team from Big Cat Rescue. We also do road signs and chicken. We also have the chicken depredation, where the approach we've been having now, that is becoming most successful, is compensation. Here, especially here in Mirador, they reject the building of hen houses. So, but instead of building the hen houses, which would be actually a bit expensive here, they prefer to be reimbursed when cats depredate their chicken. We have local people from the community responsible for checking that, and it's working so far. We have also applied this in a few other sites, but this is something that is still building up. Roads, we noticed that it's becoming a higher and higher problem, especially in the area of the Savannah tiger cat, because in the area of the Claudio tiger cat and Atlantic forest tiger cat, which are in most populated places in Colombia and Costa Rica and southern and southeastern Brazil, it is a problem already. So it's like we expanding this part as we go by. In another part that is booming now with us is the rehabilitation and release of the animals. We have two main facilities, one in partnership with the CETAS, which belongs to IBAMA, a Brazilian wildlife agency, in São Luís, where we have now 30 small cats being rehabilitated for future reintroduction and monitored, and the other one at Felinos do Aguaí Institute, where they have their own facilities and also do the same. We have people working with this. We follow strict protocols. In here, you will see like cameras that monitor the behavioral training. And once the cats are deemed fit for reintroduction, they get a radio collar and get released. Here is a release of Margay that was later monitored. Sometimes they have problems in finding the animals, but we're trying to get and this part is also beginning and let's say we're trying to expand it, but it's more expensive because it requires either GPS or satellite radio collars, and that's a bit costly. But this is something that we're targeting and believing we will expand it and have really good results, because you release the animal, so what? What happens? Nobody knows. And we need to know how effective releases are, if they are effective at all, or if they're just waste of time and money. So this is something that needs to be. I gave you just an overall thing on the conservation part, on the action taking part, and now let's see some of the science, conservation oriented, as I call science for conservation. One of them focus highly on the threats, on the invisible threat, as we call diseases. Here you see like how many dogs within and on the outskirts of Mirador State Park tested positive for canine distemper, which should actually be called carnivore distemper because it affects all families of carnivores, and for leishmaniasis. Within the park, it's like 84.4% positive for distemper and 71.1% for leishmaniasis. Outside the park, the numbers are actually a little bit better. It's worse within the park for some reason. And it's given most likely the health issue, and people are really poor and they barely can feed themselves. So the health situation of their dogs is a little bit worse. And this affects the survivorship of the Savannah tiger cat, which, if we can manage disease outbreaks to a minimum level, the population of Mirador State Park, even in total isolation, would be self-sustaining. But if disease outbreaks are even moderate, the population go extinct. So it's like we use it as our insurance policy, as I say. And we try to get this as much as possible for other ones. And the One Health approach that we've been doing now, these are for the 30 animals that are being held in at CETA, IBAMA's facility in São Luís, with our partnership with them. We're testing for FEV, canine distemper, parvovirus, coronavirus, and leishmaniasis. And the findings were a little bit scary at first, because one thing, the way we treat it, we approach it, are not really effective, let's say. I'm not saying that what we do, what it's done in general by project, but our surveillance showed that 78% of the animals we tested so far which I think were 19, tested positive for canine distemper virus. But the thing is, they had the antibodies. We need to go to the molecular confirmation to make sure. So this is one aspect that most projects may put aside, but molecular confirmation is really necessary. Otherwise, you're risking the health of the environment into releasing animals that were held in captivity. And these animals we're working with were all apprehended on trade. They were all apprehended by authorities. So they showed contact with CDV. So this is something that we'll still be dwelling on. This paper on the Savannah tiger cat made a change on its status assessment because it showed that it does not use open savanna formations, and that switched considerably its area of occupancy, reducing it considerably, that put it in a category it would not fit if we would be considering the broader habitat in general. So that's why I say science is needed for conservation, for proper conservation action. In the map you see behind, in the corner, shows the impact of dogs within Mirador State Park, showing that dogs are affecting 81% of the area of habitat available for the tiger cat, Savannah tiger cat in Mirador State Park. Another work of ours, there was actually a work of the entire TCCI team, was the when we presented a new species to the world, the Claudio tiger cat. We were 41 authors in the paper, and this paper reached huge success. It has over 28,000 visualizations according to Nature's website, and we already have 21 citations. But there are more species of tiger cats to come. So hold your horses. I cannot talk much now, but just beware, the numbers will increase. In the conservation aspect, because we not only presented a new species, but we also showed how much of their habitat they have already lost. For Savannah tiger cat, almost 56%, 64% for the Claudio tiger cat, and 68.2% for the Atlantic forest tiger cat. These are massive reductions. This changes, and we also reshaped entirely the distribution maps that was previously considered for the Leopardus tigrinus complex in general. For the vigilance, for the monitoring part, we have 30 sites for ocelot, 26 for margay, 29 monitoring Girundi, 14 for Claudio tiger cat, 14 for Savannah tiger cat, 14 for Atlantic forest tiger cat. As you see, we actually have more ocelot, margays, and Girundis on our sites because tiger cats are more restricted to some biogeographical areas, and the other ones are broadly distributed. And also, more secondarily, for the pampa cat, Cerrado black-footed cat, and Jeffers cat. And here is the results of some of our monitoring. In São Francisco de Paulo National Forest, we've been monitoring since 2000. That was actually before the TCCI came to life, but since 2004, it became part of Wildcats Americas, Gatos do Mato Brasil Americas, that Axel was trying to pronounce it earlier. So Wildcats Brazil Americas. It was within Brazil, but then we expanded to Colombia, and it no longer made sense to talk about Wildcats Brazil. So it started as a project, and now it became a conservation program that has been running since 2004. And just to show you some stuff, population drops. Here, the one on the right is at Mirador. There was a canine distemper outbreak just outside the park borders in an area we had not vaccinated before, that dropped the population considerably, but now it is recovering. Also, the monitoring contributed significantly to the red list assessment of the jaguarundi that has been just finished and is in the process of being published. And the jaguarundi changed from least concern to near threatened. Given this, you see the red line here on the chart. This is what we call the rarity line for camera trapping. As you can see, in most areas, jaguarundis are rare. They are pretty much everywhere, but everywhere they are in really, really low numbers. And it's generalized. Doesn't mean, except in very few spots, for sure. But overall, they're not the number one ranking cat in any area. Ocelot usually are. And one of the key things we're doing in the monitoring part, and here you will see like firsthand the first data known for the pampa cat, Leopardus bracatus or Leopardus labia. I forgot the name. The other name, which is also where the species is also known for. So far, we have 222 independent camera trap records. We managed to radio collar and monitor one male and one female, the male with Theralite collar, which is still working as of this very day. It's been running for 19 months, sorry. The female was, but in any event, we got like huge numbers of data points. And the area of the male was huge, over 100 square kilometers. The female was smaller, around 5. But what these two animals showed us is that they are very habitat specific, very habitat specialists. They use specifically only one kind of habitat in the Pampa biome. And at a first assessment we did, we thought 42% was lost between 1997 and 2021. However, after we got the animals monitored, the area, if you consider the area that this was done through modeling, but if we consider the habitat specificities of the animal, the area reduces to only 5%, and that includes Brazil, Uruguay, and Argentina. And the total population of this cat is expected to be between 200 and 250 animals globally. So this is a critically endangered species that we will be targeting hard to try to save it. Other issues, like we generated several density estimates for the small cats that has helped everything. Modeling, we use those densities as well to model. We model ocelot density and saw that it did impact us, the small cats density as well. Here, the data we got from the tiger cats, radio collar tiger cats, that show that a 2.1 kilo small cat roaming over areas of 60 square kilometers, 60. You didn't hear wrong, 60. So that shows how huge areas these animals need and how lower their densities actually are. And here, just some of the data we're still working on that we originally wanted to present here from some of our projects. These are not non-published data yet from several of our partners, our members, and in several areas. Since I've run out of time already, I'm just gonna show them quickly. And to end, I would just say that asking the question in the beginning, conservation actions and science can and should go hand in hand. This is the TCCI team. And in here, I end my presentation. Thank you.
Well, that is absolutely wonderful, Tadeu. And I like how you started with the conservation actions and then got to the science. So that was a very interesting, amazing, package and overview. And I'll just wait for a couple questions to come in, from the audience. But I have one for you, actually, as you so nicely sort of set the stage for science and action, how they need to go together. I'm affiliated at universities in 3 countries. And one of the things is, I think for a lot of academics within their own universities or departments, they get evaluated or rewarded based on how often they publish, how many citations they have, and how much money they bring in. And so in general, I've been observing that ecology in general is going down, whereas molecular type work is going up because it's more frequent to be able to publish and such. And then within ecology, I think that endangered species are really inconvenient because they're hard to find at low numbers and it takes a long time. And so, is there a disconnect between the academic measures of success and the measures of success in terms of applied work for conservation?
Yes, most definitely. But the thing is, I start pointing out to people, and I actually have converted some academics into start working with conservation action because, and when they start doing it, they see how wonderful it's like a self-realization thing. Actually, you have no idea how everybody described the same sensation the first time we did the intercontinental vaccination campaign, all at once. It's like it's really emotional, and it's like there's nothing that can pay for that. I used to tell people that I actually get emotional as of now. It's like the cats gave me a career. Now it was time for me to return to them. So it's like they are my bosses. I work for them, and that's it. I mean, it's commitment. I came from the old school of conservation from the University of Florida. So it's like it's ingrained in my pores, like conservation. I cannot dissociate myself with conservation. It's like it's ingrained. It's like it's part of my DNA. And what I try to do is to show people how important that is. Listen, you can do your studies, but try to also do some conservation action, as minor as they are. And and things are going bit by bit, they will. But you're absolutely right. What academia rewards is like the amount of money you bring, the amount of citations you get. I mean, productivity, how productive you are. And productivity, it's like the AG Scientific Index ranks scientists around the world based on the number of citations your papers get. So,
Yeah.
okay, that's a metric, but what have we been contributing to Mother Earth? This is something
Exactly.
that we have to appeal for. I would say like for the spiritual part. I don't know.
Yeah. Well, and it's interesting. I'll go to the next question, but sometimes I say that in our undergraduate education, we take people that are emotional, passionate, and sort of spiritual, and we teach them science. And sometimes some of that is lost actually because we teach them to be objective, to be systematic. There are a couple of questions. Vittoria Barbosa, would you like to ask yours? If you just unmute, Victoria.
Yes.
Yeah.
Yes. Thank you, Axel. Thank you, Tadeu, and congratulations for all your work. I truly admire. And could you please comment on the hybridization between Gutulus and Tigrinus, if is that common, and in the places that they occur very close environments, like in between the Cerrado and Atlantic forest, how does the identification via camera traps works for you?
It's like I've seen so many photos of these cats. You have no idea. I mean, over 10,000 of all small cats put all together. It's like even by the tail, sometimes I got to the point of knowing the species. But the thing is, Tigrinus and Gutulus, they the Pampas cat, the Cerrado Pampa cat, that I call now the black Cerrado black-footed cat because it is. Oops, my internet is unstable. Did you hear?
Yeah, it froze for a second, but we're still with you. Yeah.
Okay. I was freaking out about the instability of my connection. I had no idea.
No, it's
But I
absolutely
mean,
fine.
thanks God it worked. Uh, Getting back to that. But Tigrinus and Gutulus, we don't have any proof of their hybridization anywhere. But the thing is, it's tricky to identify small cats. That's why I've seen a lot of mistakes in papers published, even in big journals, misidentifying the species. And as I've seen them in museums, in photos, alive, you get, as I say, I have an algorithm in my mind that once I see it, it's kind of easy to say. It's like when I start receiving camera trap pictures from the Andes, I got them. That's how I got said, this cat is not on it is supposed to be a tiger cat, but it's not what we got down in Brazil and in the Guianas. This is totally different. And, it's just by knowing that, after seeing like a sequence sometimes that Reconyx cameras take like 5 shots, the animal moving, and it's like you could see the different angles and stuff, and everything like 600 pictures, your brain start like building up the archetype, and that
Yeah.
makes it easy. And
Yeah.
I've even flew from Bogota to Costa Rica just to see one cat, and I said the oncilla there is the same as in Colombia, no different whatsoever.
Yeah.
But tiger cats, especially the Claudio tiger cat, looks amazingly like a margay, and
Amazing.
they get confused. And
I'm
it's
just
like
interjecting.
it is confusing.
Tadeu, just as you're talking about that, just it's a nice segue to a question that Nadia Bao has. Nadia, please go ahead.
Hello, everyone. Hi, Tadeo. Thank you for
Hi.
your talk. Do you hear me?
Yeah, I
Yes.
can hear you,
Okay.
but I
Well,
only see a cat.
what?
I can hear you, but I can only see a cat in the picture.
Yes. I don't know how to turn on my camera. I don't know if I
Okay.
can. But if you hear me, I talk to you. Well, it was an amazing talk. I have a course with you some time ago and it was
I
amazing.
remember you. I remember you.
Thank you. I don't know if you remember me. Well, I wanted to know how do you estimate the population size of the Pampas cat, and if these 200 animals is it for Brazil
No,
or
it's
for all the
global.
range?
Global.
And
For
how
Brazil, Argentina, and Uruguay.
Uruguay?
What was
And how
done?
do you,
Uh,
uh-huh.
let What we did, based on the radio tracking data, we realized the animal uses specifically one kind of habitat, not what we originally thought it would be using. So it's more restrictive. The area is more restricted than what we originally thought of. And if we consider only this kind of formation remaining in the pampas, which is pretty much gone, what remains in there available for the animal is about 5%. I mean, this is a rough estimate, but that's so far that's what the estimate is, and based on the home range that we have, and also the camera trap data that we will be publishing now, the densities and stuff. We got to this rough estimate. This is the first estimate. We're still working on it, polishing it. But so far, the population estimate, and actually, Nadia, it's larger because before we thought it would be, at least in Brazil, 50 to 100, and maybe the same for Uruguay, which is now it's actually a little better. And we monitor the only population we know of this cat. And we have so far identified 13 individuals and 2 kittens. Well, at least there's hope because it is breeding, but it is highly affected by roadkills because the environment it lives is pretty much associated with the roads remaining in the roads that crisscross the pampas.
Yeah, thank you. Thank you, Tadeu. We could go all day because there's so much interest and so much enthusiasm, I think, online here. And also there's more questions that we can't get to, but you can see them in the Q&A. And so if you don't mind sort of writing back or in the chat, because we'll have to move on. But a huge thanks to you, not only for this great presentation, but in fact for on the one hand, quality of science, but being action-oriented, and for your passion and emotion to make a difference. That's very inspiring for us all. Thank you so much.
For inviting me to this.
Yeah, my pleasure. With this now, we're going to move on to our moderator for today from our small cat team. I'm very happy, proud to introduce Jolene Broadfield, who first joined Panthera 8 years ago, managing leopard monitoring surveys in South Africa, where she spent much of her professional career before quickly expanding her role to support the development of Panthera's in-house data systems, strengthening the organization's growing data infrastructure. Over time, she then went through this evolution, many of you will see it positive, of going to the small cats, where she now works as a data scientist, collaborating across Panthera initiatives and with many external partners. She analyzes long-term data sets to better understand small cat distribution and ecological patterns across landscapes more commonly studied through big cat research. Outside of work, this introverted, free spirit, now based back in her home country of France, enjoys rock climbing and seeks every opportunity to escape in the mountains. Welcome, Julie.
Great. Thank you so much, Axel, for handing over the baton for today's moderation. Hi, everyone. I'll do my best to guide us to the finish line, and I apologize in advance for the French accent. As Axel mentioned, the past couple of days of the Small Cat Symposium have been truly incredible. Today's talk will take us on quite a journey again. We have speakers from Turkmenistan, Chile, South Africa, project ranging from the mountains of Nepal to the highlands of Ethiopia, all the way down to the sea level in Trinidad and Tobago, all without any additional CO2 emission, by the way. So this again illustrates the remarkable diversity of the small cat world. I'm confident this presentation will give us a high note to finish on. And with that, we'll start our speed talks session, which is, by the way, a format we added in order to include as many presenters as possible in the symposium, considering the breadth of amazing abstracts that were submitted. So short for sure, but still a great opportunity to share an overview of those amazing projects out there and spark connection, hopefully. So with that, we'll start with our first speak talk of the day with Aknabat Potaeva. She is a lead scientist at the Kopetdag State Nature Reserve in Turkmenistan, studying and preserving the reserve unique mountain ecosystem and wildlife. In her role, she also leads research, monitoring, and wildlife conservation efforts across the country. I believe she's not online, so I think we're going to play a recording for her.
Good morning. The cold winter deserts of Turkmenistan form a globally unique but still poorly studied ecosystem, located at the crossroads of temperate continental and arid biogeographic zones. Our more than a decade of camera trapping sign surveys and field observations conducted both inside protected areas and across surrounding unprotected landscapes. We assess the status, distributions, threats, and conservation needs of wild cats in these cold desert systems. Turkmenistan is home to 6 species of small cats in lynx, caracal, manul, jungle cat, Asiatic wild cat, and sand cat. One of our most significant findings concerns the caracal. After 50-year gap, we recently had a new detection in Badri on the border with Iran and Afghanistan. From a handful of records we obtained in the capital and Balkan regions in 2025. This extreme rarity reflects the mammal's high sensitivity to human disturbance, habitat degradation, and predation pressure. Sand cats are similarly elusive. A rare observation from the Karakum Desert confirms their continued presence, but the species remains among the least recorded felids in Turkmenistan due to low densities, nocturnal behavior, and reliance on remote desert habitats. Across all species, we identified dog predation by guard and hunting dogs as one of the most important sources of mortality, particularly for manul and sand cat. In addition, Asiatic wild cats face increasing threats from hybridization with domestic cats, road mortality, and occasional trapping by herders. Many of these pressures operate outside protected areas, in buffer zones and ecological corridors where enforcement is weak. Protecting small wild cats in Turkmenistan winter desert requires moving beyond site-based conservation alone. Priority actions include expanding protection to key landscapes, maintaining ecological connectivity between habitats, reducing dog-related mortality, and strengthening community engagement and monitoring beyond reserve boundaries. Thank you.
Great, thank you. Unfortunately, she's not online, but if you would like to reach out to her, please don't hesitate. We have on the website a book of abstract with every presenter's contact details. So next we have Badri Baral. Badri is a Nepali ecologist and biodiversity conservationist serving as executive director at Nature Conservation Initiative Nepal and a member of the IUCN Small Carnivore Specialists Group. His expertise span high-altitude landscape ecology, carnivore ecology, community-based conservation, biodiversity outside protected areas, and conservation policy research. Thanks, Badri, and welcome. Please take it away.
Thank you. Hello, greetings of afternoon. Today I'm going to present range expansion of Asiatic golden cat. And as you know, Asiatic golden cat is listed as vulnerable on IUCN threatened species category, red list, and it is listed as data deficient by Nepal red list. Right now, Nepal government is revising its. Nepal government is revising its red list category, And among 13 wild felid species of Nepal, Asiatic golden cat is one of the least studied and overlooked species. And methodology. We have been awarded with Rupert Grant for red panda conservation in the Jazakot districts, and we have trained a total of 9 citizen scientists and deployed them for camera trap survey in order to monitor red panda in Antia, which is located outside the protected area. After having completed survey, our team unexpectedly captured Asiatic golden cat. Its presence was subsequently confirmed by experts after systematic review. And from more than 59,000 photographs, over 1,027 camera trap days, a single image of Asiatic golden cat was captured in Jazakot temperate forest. And the photo was identified as Asiatic golden cats from expert consultations by its unique traits. And this is the first capture in Jazakot district. It is the crucial western habitats in the country, and it extends its known global western range by 395 kilometers. And there is need for increased survey reports in Jazakot, as it is only from bycast data, so that we need to have increased survey reports in the Jazakot and adjoining area so that we have better result. And its conservation impacts. It unlocks critical opportunities for research and conservations. And this record may be potentially maybe the opportunity for collaboration to work with community-based conservation, as we have been doing with Panthera SCF, and the project is ongoing right now. And geographic significance and the habitat. This discovery extends the non-western limits of Asiatic golden cat by approximately 395 kilometers west of Gauri-Shankar Conservation Area, suggesting its potential habitat in between and connectivity towards Jazakot. The cat was found in diverse mountain forest characterized by fir and rhododendron. Here is the map illustrating its range expansion from Gauri-Shankar Conservation Area to Jazakot, which is located outside the protected area. And so we can collaborate with this regards for community-based conservation community-based conservation. And finally, I'd like to thank Nature Conservation Initiative Nepal, my organizations, NERI and Panthera, Claudio Report Working Groups, Rupert Foundations, University Grand Commission, Idea Wild, Gangaram Regmi, Yadav Gumire, Wilcox, and Ming. Thank you for collaborations and potential collaborations. You can contact me in the mentioned address. Thank you.
Great. Thank you very much, Badri. I'll let the audience send in the questions to the Q&A tab. But yeah, thank you again for this lovely talk on a fascinating yet understudied species, and quite timely as the Asiatic golden cat was recently reclassified from near threatened to vulnerable. This range extension is definitely an important finding. Thank you, Badri.
Thank you.
I'm gonna see in the Q&A. We have a question from BJ. BJ, if you'd like to unmute yourself and ask a question or comment.
Yeah, hi everyone. This is Vijay from Nepal. Yeah, very exciting to know that the work outside protected areas from Nepal are bringing up the new data sets for those species that are considered as least concerns or data deficits in the red list criteria. And I just wanted to congratulate Badri and his team for this tremendous success. Yeah, thank you.
Thank you, Vijay.
Thank you very much to both of you. Great. So next we have Bello Adamu Danmallam. I'm hoping I'm pronouncing it right. He is a conservation scientist and data analyst with a deep passion for wildcat species and their conservation. For the past 2 years, he has served as the Species Survival Coordinator with the IUCN Species Survival Commission Center for Species Survival Nigeria, based at the AP Leventis Ornithological Research Institute. working through the assess, plan, act network communication cycle to drive evidence-based conservation outcome across the region. Thanks, Bello, sorry. Please take it away.
All right. Hello, everyone. I'm delighted to present our analysis of the drivers shaping the distribution of the African golden cat. All right. The African golden cat is one of Africa's least known felids, elusive, and still poorly understood. Yet it occupies some of the continent's most threatened forests and is highly vulnerable to increasing human pressure, including habitat destruction and hunting through wirelessness, which are usually set for antelopes, but they catch African golden cat too. However, we still lack a continental scale understanding of where suitable habitats remain. We compiled 309 occurrence records from various sources and integrated them with environmental predictors representing forest cover, climate, and human pressure. And to prevent inflated predictions, we restricted model calibration to the Guinea, Congolian, and Afro-Mountain forests, excluding known barriers such as the Dahomey Gap and urban areas, urban pixels, before projecting at a continental scale. Without these restrictions, models may predict suitability in ecologically inaccessible regions like deserts or open savannas, or even cities. So we then use ensemble modeling approach combining in biomod 2 package in R using generalized linear models, generalized additive models, gradient boosting machines, and random forests. We found that 3 predictors structured habitat suitability, together accounting over 80% of total variable importance. Distance to protected areas contributed over 30%, with suitability declining sharply as distance increased. Precipitation seasonality showed a unimodal response with peak suitability at intermediate levels of precipitation seasonality, which likely reflects prey dynamics. Forest cover increased suitability up to about 85% canopy cover before declining. And this decline likely reflects reduced detectability and limited surveys in very dense forests rather than through ecological avoidance. Our ensemble model estimates over 600,000 square kilometers of suitable habitats across Africa, while protected areas in green encompass 53% of this area. Nearly half, about 47%, lies outside formal protection. All right. Protected areas clearly function as critical refugia for the African golden cat because they protect African golden cat prey base and limit snaring to some extent. Therefore, conservation must prioritize improved protected area management, connectivity between these strongholds, maintain forest corridors, and manage land pressures even beyond park boundaries through community-based approaches. Building on this work, we have deployed camera traps in Nigeria's largest national park, the Geshaka-Gumti National Park, to generate critical data to inform conservation planning. At the same time, we are working closely with local communities to assess their knowledge of the African golden cat and advise them on how to protect the species. Well, the long-term survival of the African golden cat depends not only on protected areas, as we've seen, but on integrated forest conservation and community-based efforts across its range. We acknowledge Panthera for their crucial support through the Small Cat Action Fund and our collaborators and local communities who made this work possible. Thank you very much for listening.
Thank you, Bello, for this excellent presentation. We definitely need to connect. While people are typing their question, I was wondering, is there plans for those findings to help guide future survey efforts for African golden cats and within the local research community?
Absolutely, Julian. Thank you very much for that wonderful question. So we currently have cameras deployed in the national park, use the habitat suitability modeling to inform our camera trap placement. And we're also working closely with the local communities. And as far as we know, this national park, the Gashaka-Gumti National Park, is the last remaining stronghold of the species in Nigeria. So, and the data we're generating from the camera trap surveys, we believe will be very instrumental to inform national conservation planning, as red list assessment, the national red list assessment, which is we have plans for that. So whatever data we get from the national park is our national population for the African golden cat. So absolutely, there are long-term plans after this work. Thank you.
Great. Thank you, Bello. This is amazing. We'll now move on to our next talk, which I'm very much looking forward to. We next have Tyler Murray-Ramcharan. Tyler is a DPhil researcher at the University of Oxford, working at the intersection of astrobiology and conservation, exploring how tools developed to detect life in space can be applied to monitoring elusive species on Earth. Alongside his work, he leads conservation projects in his home country of Trinidad and Tobago, focused on camera trapping and community-based research to support the monitoring and protection of ocelots. Tyler, thank you and welcome. The floor is yours.
Can we all see the presentation?
Yeah, perfect.
Okay. Thanks for having me. Today I'm going to try and give a quick flavor of some of the work I've been doing in Trinidad with ocelots. So no space stuff today, unfortunately, but this should give you an understanding of what's going on with Trinidad's ocelots and what we're doing to better understand and protect them. So to start things off, this is the range map from the IUCN Red List for ocelots, and Trinidad is located there. It's a dual island nation, Trinidad and Tobago, and we're the southernmost Caribbean island located just off the coast of Venezuela. I'm speaking about Trinidad today because Tobago does not have any ocelots. And by the looks of this map, Trinidad does not have ocelots either. So we're currently not on the range map. And this is likely because we have nothing published about our ocelot population. Well, that's one reason, but I actually think it's because our ocelot population is quite special. So hear me out. Nowhere else in the world really will you find an isolated, long-standing native island population of ocelots that's been separated from the mainland for 11,000 years. They're also our apex predator. There's no jaguars, no pumas, nothing larger. They're the top cat on the island, and they've been serving this function for thousands of years. We also have some indications that they could be different to the mainland species, namely the size, the shape, the coloration, distinction in terms of spot patterns, and most notably the coloring of their noses. Trinidad's ocelots have black noses mostly, whereas their cousins across the water have pink noses mainly. In Trinidad, they're threatened primarily from the loss of habitat from land conversion, which is happening quite rapidly, wildlife conflict, hunting, poaching, the pet trade, and stray dogs, which not only bother them, but have the possibility of transferring disease. Even though globally they're listed as least concern, they're listed as environmentally sensitive in Trinidad, so they're afforded the highest possible protection. Now, what we know from previous surveys, there has been some effort here, is we know roughly where they're located on the island. And then most recently, we've done some surveys to try and get density estimates. The only one that we have a density estimate from is this one, the northwestern tip of the island. And we conducted a camera trap survey in 2020, and we were able to get quite a decent amount of pictures of ocelots, but a rather low sample rate. And we were able to catalog 8 individuals and get our estimate, which was roughly around where it was expected. We also looked at overlap, which was high with red brocket there, low with agouti, and we found that they occupy the entire peninsula. And there were some severe limitations with this study, including our low recapture rate, the low sample size, which means these estimates should be taken as precaution, but this paper is in preprint and hopefully will be published soon. In terms of what we don't know about ocelots, it's quite a lot. So we don't know how many there are left on the island. We don't know how their population's changing. We don't know what they're eating and how that diet changes across different local subpopulations. We know generally where they like to be, but we don't know what habitat suits their preference. And whilst we know that they're important, we just don't know how important. And finally, we don't know in terms of their genetics if they're distinct from the mainland population and what this means for their long-term viability. So quite a lot of work to do. So Literally a few weeks ago, we launched this project, the Trinidad and Tobago Ocelot Project, which main goal is to create a coordinated long-term framework for effectively collecting the information we need to properly assess their status, the habitat use, and tackle the threats that are facing them. And the phase one of this begins in the northern part of the island with 2 study areas. And the objectives here are to do coordinated community-led monitoring, meaning building capacity for the community to undertake the camera trapping, establish meaningful baselines for comparison, so doing this every year going forward to see how the population is changing, and directly on the ground tackling threats. We started things off with a workshop. This happened 1 week ago, and we had a huge turnout. Over 60 people came down. We spoke about ocelots, we spoke about the project, we got hands-on with camera trapping, as well as some of the tools that we're using to analyze the data, and we had a huge volunteer sign-up. And then we also launched our social media, which is getting a lot of attention, and we've even gotten some local news attention for the project. So there's a big passion from the community around this work. We also put out small cameras, and we're doing that's currently running. And then we are planning to do the second survey at the second site later this year. The intention behind this is to do annual monitoring, expand to more areas of the island, eventually collect the data we need for suitability modeling to get that island-wide estimate, and then start tackling the threats, whether that's securing chicken coops or tackling the transmission of disease from dogs with vaccines or neutering. We also have a local ocelot documentary that is in progress. It's in the very early stages, but we're looking to get some more attention for our unique species population of ocelots that we have on the island. And we're partnering with more regional contacts like the Ocelot Working Group. I'm exploring the possibilities of supporting the project going forward with more local funding through the interest that we've been gathering on island. So this is only the beginning of the work that's going to happen. There's lots to do, but we're very excited with all of the enthusiasm we've gotten from the community. Just a quick thank you to all the supporters of the project, both international and local, and the Rufford Foundation, who is currently funding phase 1. And yeah, that's all. Thanks for listening. You can contact me if you're interested or have any questions.
Thanks, Tyler. Amazing. Fantastic to hear more about this island population. I'm going to see if we have question. Not yet. So I'll jump on one. To what extent is the government engaged with or supportive of your work on ocelot?
The support has been fantastic, and actually they're the ones providing all of the equipment and camera traps,
Wow.
all the permits, and so that we're really working together with everybody on the island. It's been a massive joint effort, which is the first time this has happened. So yeah, very encouraging.
Yeah, that's amazing, especially from the start. It's great.
Yeah.
Great, thank you very much, Tyler. I'm still going to look. We do, I think, have a question. Sugoto, do you mind unmuting yourself and ask a question?
Yeah, I believe that Trinidad has tyras as well. Do they outcompete ocelots? Do they fare better under habitat threats and things that ocelots are facing?
I believe they do quite well, and we have some evidence from our camera trapping the island where we've seen ocelot numbers declining and then the tyra increasing. In terms of are they outcompeting food sources and so on, I'm really not sure, because we have some evidence to show that the ocelot's diet in Trinidad is so varied, from monkeys to possibly juvenile deer, agouti, rodents, and the tyra maybe a bit more niche. So yeah, we're not sure, but it's definitely something we'll look into.
Great. Thanks, everyone. Thanks for this great question. And thanks, Tyler, for this amazing presentation.
Thank you.
Great. Our next presenter has been very active in the discussion, asking many thoughtful questions. So I hope the audience will do the same for him. We have next Nicolás Gálvez. He is a researcher in carnivore conservation and coexistence in agriculture landscape in southern Chile. He contributes to knowledge and conservation of the guinea over the last 20 years. Thank you and welcome, Nicolás, as a presenter this time. The floor is yours.
Okay. Let me get my stuff together. I thought it was going to work, but it didn't. All right, so hello everyone. Today I will talk about the potential of zenith camera trap placement using the Winya or Cod as an empirical example. So zenith placement refers to positioning cameras vertically to capture the dorsal pattern of the animal. The Winya is the smallest wildcat of the neotropics and has a subtle and non-conspicuous pelage pattern. Previous work has shown that the dorsal area displays high variability among individuals in controlled conditions. So we asked ourselves, is it possible to recognize individuals using the dorsal patterns under field conditions? We recently conducted a review of studies involving 9 small wildcats of South America. For the Winya, we found that despite extensive survey effort, there are no density studies based on individual recognition. This highlights both the challenge and the need to develop such methods. The species was recently downlisted to least concern due to improved information. However, this data relies heavily on conservative values from telemetry or detection rates, which is not ideal, specifically for the more critical geographic regions. Therefore, density estimates based on individual recognition would provide valuable data for future assessments. In our study area was located in the Reñué Conservation Area, in a very remote area deep in the temperate rainforest of southern Chile. In our methods, we propose several field considerations that we believe are important to evaluate for any species when using zenith camera traps. We applied a series of inclusion criteria for images to be used for the recognition. So first, we filtered images based on visibility, so how clear the dorsal area was exposed, and sharpness. Then we edited images to remove the background and increase the contrast. And each dorsal pattern was given a unique ID linked to its event and site. Then for identification, a human evaluator searched for at least 2 diagnostic marks without knowing if the photos were from the same event. And then accuracy was assessed by verifying if the identified markings matched images from the same event. Here you can see some of the examples of many of the diversity of the patterns that we obtained. And so after applying all filtering criteria, we analyzed 320 photos from 210 events and recognized 12 individuals. Importantly, 100% of the dorsal images were correctly matched to their corresponding event. Here's an example of some of the identified individuals with some of their diagnostic marks that were identified, specifically at the base of the head and the base of the tail. So what key takes do we have? Basically, individual recognition from dorsal patterns is possible. There's an advantage because the field of view of the camera is accurately estimated, and so could be useful for random encounter models or photogrammetry. Only one camera is needed for individual recognition, which could have implications on survey cost. There's definitely a potential for AI use in identifying individuals rather than a human evaluator, for sure. Perhaps other species may possessing distinct dorsal patterns this technique may be useful, and definitely there's an application to further improve the density estimates of the Winya. Here are my co-authors. This work is currently in press in Oryx Conservation Journal. Here's a QR code where you can access more of my work, and here's an email if you have any further questions. So thank you very much.
Thank you, Nicolás. Sheesh, that's an excellent work. I'll give people a moment to submit their question, but it's especially exciting to see cost-effective method like this and thinking out of the box. I really love this this presentation. Thank you very much.
Thank you.
I'm gonna
Thank you, Jolene.
I'm gonna see if we have any questions in the Q&A. Not yet. I just spoke too quickly. We have a few. Rue, would you mind unmuting yourself and ask your question?
Yeah, that's really interesting technique actually, and I can see it working for wildcats in Europe and in Scotland as well. I have one question though, and well, we find that detectability in the first place can be very low, and so we often kind of, I guess, rely on a wide field. So I know it's maybe less statistically rigorous. Did you compare detectability with that camera setup versus a horizontal setup?
Yeah, that's a great question. And unfortunately, we had some field limitations regarding having basically, because we would need to have the zenith camera and both of the lateral cameras to kind of test that. But we did evaluate, and it's in the paper, we evaluated detection rates. So not probability detection, but rates of detection, number of photos per time. And we compared them with other published studies for the species, and they were quite comparable. They're not much different. So
that's positive,
in that sense, it's positive, right? Yeah.
Or you're just very good at finding them. That's your camera setups.
Right. Yeah.
But yeah, that sounds good. Thank you.
Thank you.
Thanks, Rue, and thanks, Nicolás. I see there's another question, but unfortunately, for the sake of time, we need to continue. Would you mind, Nicolás, maybe popping an answer on the Q&A?
Yes.
Tab? I appreciate it.
No worries.
Thank you very much.
Thank you very much, Yoli. Ciao.
Thank you. Great. I think next we'll take a 5-minute break. Stretch your legs, have a rehydrate, and come back ready for more fascinating small cat science. I'll see you shortly. Welcome back, everyone. I hope you had time to have a little stretch. We're now going to jump into the long talk sessions, and we will start with Michelle Schroeder. Michelle is a terrestrial ecologist with extensive experience working with small carnivores. She joined the Black-footed Cat Working Group in 2019 as a telemetry field monitor and initiated a collaborative, non-invasive genetic research project using detection dogs and molecular scatology, now the focus of her PhD at the University of Cape Town. Michelle, thank you and welcome. The floor is yours.
Yeah, I've been working with black-footed cats pretty much just with my canine field assistants to keep me company. So really appreciate the opportunity to finally meet up and share with the small cat community. So thank you. And you guys have already been introduced to black-footed cats with some great presentations. So move on. And yeah, we know a surprising amount about their ecology from the Black-footed Cat Working Group's telemetry sites. And a variety of threats that I'm sure you're all familiar with have been identified. Potentially compounding these threats, though, whole genome sequencing has revealed that they have low genetic diversity from repeat historical bottlenecks and carry mutations, predisposing them to deadly diseases. So this can reduce adaptive capacity and leaves them more vulnerable to the negative impacts of small isolated populations. So this is particularly concerning because climate change is predicted to disproportionately impact the Namakuru. However, the samples mainly come from captive individuals. We currently lack any contemporary genetic diversity and population level data across the species range. This is largely due to not really having an effective survey method, but camera traps don't really work well with them since they don't use predictable paths and they're obstructed even by short vegetation, so resulting in few camera trap records throughout their distribution. Alternatively, SCAT switches the focus from capturing a species at a specific location to locating evidence associated with their natural behavior and movement patterns. SCAT is also a powerful source of genetic data, allowing us to identify individuals and estimate populations and get insights on important genetic health indicators. The challenge, of course, is finding these little tiny scats across huge landscapes. So that's where the dogs come in, sniffing out what we can't see. Visual surveys are more prone to overrepresent dominant species or individuals. So by relying on olfaction, dogs can reduce potential status and sex-based sampling bias. Plus, dogs are better at discriminating among closely related species, which reduces unnecessary lab costs. So I first set out to evaluate if the dogs were even effective at detecting black-footed cat scat, and whether the scat DNA could produce reliable data for identifying individuals and ultimately estimating populations. I'm not a geneticist by training, so this third objective is what I've been working on more recently and the focus of this talk. So we first applied this approach to the only existing black-footed cat study sites by the Black-Footed Cat Working Group, Benfontein Nature Reserve in Central South Africa, and Grenau in Southern Namibia. So for this, I trained Laika, a 5-year-old ball-obsessed female German short-haired pointer, to locate black-footed cat scat. So our routes followed 2 predetermined 1,600-meter polygon routes per 4 square kilometer grid. And as the handler, I would walk along the transect. Laika wore a GPS collar and searched off lead, allowing her to work odor and variable winds and increase coverage. So these images just highlight why the dogs are essential. So many of the scats were effectively invisible to the human eye. That one I still can't even see. Surveys were conducted during the cooler, drier winter months, which is better for working dog abilities and DNA preservation. So we completed 55 routes in South Africa and 105 routes in Namibia. Laika located 63 scats in the South Africa site and 123 in the Namibia site. And these are stored in silica desiccant and shipped off to the National Genomics Center in the US for DNA sequencing. So you can see that the dogs were covered at least 3 times human distance. And we first had to determine species ID, which we used 16S universal primers. So 89% of these samples had sufficient mitochondrial DNA quality to determine species, of which all 4 were from black-footed cat. So we used a specially designed microsat panel to genotype individuals, which was successful for 128 scats. So overall, the detection dog proved highly accurate, nearly 98% known target species accuracy. And our lab results exceeded expectations for scat samples with almost 70% genotyping success. So beyond population estimates, this genotyping data can also be analyzed to explore genetic diversity and reproductive behavioral strategies. So this being the first genetic study of wild black-footed cats, it provides a baseline to compare with other solitary felids. So despite the low genomic diversity at the species level, both populations showed moderately high microsatellite heterozygosity with no signal of inbreeding. The observed heterozygosity was above average compared to other solitary felids. It was most similar to the jungle cat and puma. Allelic diversity, however, was a bit on the lower end, more similar to sandcat and Eurasian lynx. And comparing between sites, the differentiation was small but significant. Only about 4% of genetic variance is attributed to differences among populations. So these results are consistent with recent or ongoing gene flow. So what reproductive mechanisms contribute to this genetic diversity? So we can use different analyses to determine which sex stays or disperses. The most reliable is sex-based spatial genetic autocorrelation analyses. then it would predict that higher relatedness among females than males, especially at the nearest neighbor distance. And this was confirmed, supporting female philopatry. Another approach is sex-based population assignment test. So the dispersing sex should show more negative assignment values, which we also observed consistent with male bias dispersal. While effective dispersal distance is also very relevant, at the scale of our studies, we were not able to detect that. So to assess the mating system and reproductive skew, I ran parentage and sibship assignment analyses in Program Colony. So it uses a conservative likelihood approach and works with non-invasive samples. So these results indicated of the 47 sampled individuals came from 15 inferred fathers and 17 mothers, which is fewer than expected under random mating. The sibship network was dominated by half-siblings with very few full siblings, which is consistent with polygamy and non-monopolized mating, where females mate with multiple males across reproductive events. However, with scat, you don't have age structure, so there are overlapping generations, which creates uncertainty in the models. But we did observe a similar pattern if we only included high probability relatives. So 16 offspring from 3 parents were identified. We had female 46, had 6 young, and 2 males had 5 and 6, both siring offspring with this female 46, which provides further evidence that females mate with more than one male, at least across the reproductive events. It also suggests that reproductive success is highly skewed. So how do we interpret these genetic patterns? The Karoo region is highly variable, has highly variable rainfall with frequent droughts, resulting in these boom-bust small mammal abundance patterns. Our Namibia surveys coincided with above average rainfall, and the telemetry data from collared females showed increased reproduction. For the subsequent year, there is drought and high mortality. So the genetic signatures make sense in a metapopulation structure with fluctuating populations. So as local populations shrink, rare alleles are lost faster than heterozygosity, and genetic diversity and connectivity is maintained through male bias dispersal. Reproductive skew and polygamy promote more rapid localized adaptation, but at the risk of genetic erosion and demographic collapse if they become isolated. So the scats continue to provide data for additional studies on diet and endoparasites, using DNA metabarcoding and stable isotope analyses. We more recently surveyed mixed livestock agricultural area. This enables a comparative health and diet study across land use gradients. While results are encouraging, this study is limited to 2 snapshots. So the broader value lies in scaling this approach range-wide across predicted subpopulations to assess connectivity and habitat suitability. We can then model the effects of land use and climate change, population size, distribution, and genetics to support conservation and land use planning. So despite a history of overgrazing, Southern Africa's semi-arid rangelands are a relatively natural state, but they're also a frontier for development, especially with renewable energy. So we have this window of opportunity now to potentially restore and or maintain connectivity before it's lost. So overall, with the help of our specially trained dog, obtaining sufficient sample sizes to better inform the species status and guide conservation efforts is now within reach. So beyond black-footed cats, dogs can easily be trained to search simultaneously for coexisting carnivores. And perhaps these methods could be adapted to support other small cat species globally. So dogs and non-invasive genetics are not without their challenges, but I hope this study has demonstrated that monitoring the genetic diversity of elusive cats is both achievable and essential. So this was in partnership with the Black-footed Cat Working Group, and big thanks to Panthera for supporting the project and many others. Thanks for listening. Happy to answer any questions.
Amazing. Thank you, Michelle and Laika, for all this work and presenting such interesting findings. While I give a moment to people to type their question, which, by the way, you can do at any point in the presentation. Huh? I just wanted to say that the accuracy rate is really impressive. Presumably, it will allow project fund to be focused on more targeted genetic analysis rather than on misidentified scats. I'm gonna jump on a question as I don't see any others yet. On the topic of detection dogs, I was wondering if you might have some lesson learned that you would like to share with the audience that could be interesting in this type of method. Very good looking tool.
Yeah, no, the dogs have taught me a lot, so it'd be hard to pick out just one thing, but yeah, you have to trust your training and trust your dog. It took about 2 years before I even got any genetic results back. Some of the scats I saw looked quite large. I did not think they were a black-footed cat, and they did turn out to be black-footed cats. So gotta trust your dog, but also trust your training.
Great, thanks. Another question would be, how do the climate conditions, especially in the area you worked, affected the quality of the extracted DNA?
Yeah, so I mean, I did surveys in the winter, which is also the dry season. So cool and dry is quite good for DNA preservation. So yeah, I think that helped a lot.
Great. Well, thank you for that. Thank you, Michelle. It was amazing. Next, we're gonna move on to our next second long talk with Marina Favarini. Marina is a PhD in animal biology and from the Federal University of Rio Grande do Sul with research focused on spatial ecology and conservation of small neotropical felids, member of the Pro-Carnivorous Institute and the founder of the Felinos do Pampa project. Thank you, Marina, and welcome. Please take it away.
Okay, so are you listening to me? Yes?
Yeah, we can hear you and we can see your presentation.
Okay, perfect. So hello, everyone. It's a great honor to be here at the Pampa Symposium. And today I will present the Wildcats of the Pampa project from conflict to coexistence. So our project takes place in the southern Brazil, in the Brazilian Pampa biome that it's in contact with the Atlantic forest. So it's a region with an incredible biodiversity. So as you can see, the Pampa biome has been experiencing increasing anthropogenic pressure, mainly associated with the agricultural intensification. So in the Pampa, we share our landscape with 7 species of wild cats. So the first is the Joffrey's cat, the margay, the jaguarundi, the critically endangered Munoas pampas cat, the southern tiger cat, the ocelot, and the puma. Unfortunately, there is still very little knowledge about their ecology. Because of the fragmentation, our wildcats face several threats. These includes also high rates of roadkills, intense retaliatory hunting caused by conflicts with local communities, and the pathogen transmission from domestic dogs. So the threat of the silence, it's like the misinformation contributes to the invisibility of wildcats. So we cannot preserve what we don't know. So our mission it's to change this reality. Our goal is clear: to mitigate this threat by actively promoting an harmonious coexistence between humans and these 7 wildcat species. 7, sorry. So to change this reality, I'm sorry. So we created a multi-strategy that includes education, roads, ecology, conflict, coexistence, and the One Health approach. And so today I will present only the conservation actions that we do. So to breaking this cycle of misinformation, we must educate. We conducted 72 educational actions, reaching over almost 8,000 people across different 26 community engaged. So our efforts also included 29 appearances on television programs and podcasts to spread the message of wildcats. So here, just to show you some pictures of our environmental education actions. We work mainly in schools, but we also work in community events, like Children's Day, Environmental Day. We also developed some games, memory games, and trail games with the Joffrey's Cat Working Group. And we also create all those materials as books, guides, brochures. So Flávia Tirelli did this little book from Pampas Cats and the Powder of the Invisibility. And a biology student, Julia, developed another book here that talks about the threats of the species that they are suffering. So the conflict predation. So the biggest flashpoint for conflict here is the predation of domestic poultry, which often leads to the retaliatory killing of the wildcats. Here I want to show you one case where a farmer contacted us asking for help because a cat was stealing his chicken. So I'll try to put a video here. So from the video, so we went to the site and installed a camera trap. And to our surprise, the cat was a Joffrey's cat that were stealing all his chickens, one by one. So there is another case that was also a Joffrey's cat stealing chickens. So from the video, it was possible to see how the cat was assessing the chickens, and we could propose closing and reinforcing the chicken coop. So the owner, of course, accepted that, and we also replaced the chickens that he missed, and we had a very happy ending with a farmer that started to like the Joffrey's cat, and the cat's still alive. So after this first case, many others began to appear. So to address the problem at the root, our team started working directly with the community and interviewed in 36 chicken coops, making them predator-proof. And as a result, retaliatory killing stopped in these properties, and we proudly certified these farmers as members of the Felid Human Coexistence Network. So we give them this member sign, and they were very proud to have this on their farm. So here's just to show another case and that we worked with. And this Joffrey's cat here, he came back to trying to have the chickens like for 15 days. After that, he gave up and stopped appeared at this property every night. So another massive threat is the roadkill. So to mitigate this, we installed 12 road signs to alert drivers. We also built wildlife crossing equipment with 1 kilometer of directional fencing. We implemented 5 speed registers, and we mapped 150 kilometers of federal highways to assess the feasibility of new underpasses. So here just show you some of our road signs and the wildlife crossing under the road, and a big billboard we did for Pampas cats. And well, we also work a lot with citizen science, that we know that local knowledge are essential for our works, as people who live in these landscapes observe wildlife daily and can provide valuable insights about the presence and behavior of wildcats. So then we created this Pampas Cats panel, and that is right, wanted Pampas cats. So we asked the community to share information about the species, and we received photos and reports describing behaviors that helped us better understand its escape behavior, that is very characteristic from Pampas cat, how is his defense. Another case was a police officer, that it's this Pampas cat here, that found this cat, and he recognized the Pampas cat. He thought at the beginning it was a domestic cat, and then he realized that was a Pampas cat because he saw the outdoor, and then he contacted us, and we confirmed that was a Pampas cat. So this individual was released in a safe place. So through citizen science, we also obtained more than 178 records of road-killed cats across the state. These records are extremely valuable because they help us identify collision hotspots. With this information, we can propose target mitigation measures for these roads. So all these points are one road-killed animal all over our states. Well, the silent killer. Domestic animals roaming freely pose a dual threat. They can hunt wildcats and also transmit deadly pathogens. So to reduce the risk and avoid conflicts with wildlife, we worked with local residents to build kennels and encourage responsible pet management. So until now, we have constructed 3 kennels in one area that we monitor the wildcats. And well, the One Health approach, we vaccinated, neutered, and facilitated adoption over 1,000 domestic dogs and cats to prevent spillover pathogens. So here's another example. One important action that we did a vaccination campaign inside the indigenous community, which is one of the last refuges for the Southern tiger cat in our metropolitan region. We also built 2 new rehabilitation enclosures for wildcats at the local university centers, such as the Preservas, that it's here in the metropolitan region, and another one in the Nerves, more through the south of the state. So this provide a safe space for injured and rescued wildcats and to recover right before being released back into the wild. So here is some cats that were had some issue and went to these places. Well, we know that to protect the Pampa and the local economy and culture, we must thrive along wildlife. Therefore, we have established partnerships with local business, including a winery, this Batalha. So they created two special editions featuring the Pampas cat on its label. And we also collaborate with the Potiguarani indigenous here, and with the association Bichos, the artisans Bichos do Mar de Dentro from Pelotas. So we believe that by supporting these local artisans and the indigenous community, we help stimulate local craft commerce while simultaneously raising awareness about wildlife conservation. So, protecting wild cats in the Pampa goes far beyond ecological research. It requires working together with local communities and recognizing that conservation only works when people are part of the solution. So, by building this partnership, we hope to create a future where people and wild cats can coexist in this unique threatened biome. So, here is our team, our big team. And we really believe that conservation is truly a collective effort, and none of this work would be possible without the support of many partners. We are deeply grateful to our local communities, volunteers, and global collaborators, including Panthera Small Cats Conservation, Mohammed bin Zayed, Big Cat Rescue, among many other support work. So thank you very much for your time and the opportunity to be here. Sorry about my English, and I'm here for questions, and this is our contacts. Thank you.
Amazing, Marina. Thank you so much. Very interesting. It's clear a large amount of work has gone into this project. While we wait for questions, I wanted to say I especially appreciated how you highlighted the importance of the social side of things, because ultimately wildlife conservation happens in human landscape, and understanding people can be just as important as understanding the species. So yeah, thank you for that. I'm going to see if we have some question here. We don't have yet, but if they do come up, please stay a little while on the QA tab. If you can answer them, that would be great. Thank you again, Marina.
Yes, sure. Thank you.
Great. Next, we have our first and only duo presenter talk. It will be a recording from Anaïs Beaumariage, who is a project manager at the Conservation Genetic Laboratory in Belgium. Her work focuses on population genetic of several mammal species, wildcats, lynx, otter, beaver, wolves, to better understand conservation action. And with her, we have veterinarian by training, Laurie Bedouet, who will be available to answer the question afterwards, and is currently finalizing her PhD on the conservation of Eurasian lynx in France and Switzerland. Her research combines ecotoxicology, genetics, and trophic ecology to address the ecological challenges that this species face in human-dominated landscape. So I think we have a recording queued up for this presentation.
Hello everyone, my name is Laurie Bedouet. I'm a PhD student from Chrono Environment Laboratory in France, and I'll be co-presenting with Anaïs Beaumariage, project manager in the GECOLAB in Belgium, our collaborative work with the French NGO SFEPM on the genetics of the Eurasian lynx in France. For the context, the Eurasian lynx is one of Europe's most iconic large carnivores. It disappeared from France and all of Western Europe in the early 20th century due to direct persecution by humans, habitat loss, and predation, mainly ungulates. Changes in public opinion and legislation in favor of lynx conservation, combined with habitat restoration, contributed to the success of various reintroduction programs across Western Europe. This map highlights some of them, showing the date at which they occurred, the number of individuals reintroduced in brackets, and their source population. Our study focuses on the Jura and Alpine populations, which originated from the reintroduction of about 20 individuals in Switzerland during the 1970s, sourced from the natural Carpathian population in Slovakia. From Switzerland, the species quickly colonized the French Jura through natural dispersal and now occupies almost all forest massif of this mountain range, as shown by this map, representing the regular and occasional presence of the species in that area. In the French Alps, The species is essentially present in the northern part with a tendency to colonize further south. It is also expanding westwards with more and more sightings being reported. Despite this apparent growth, the species remains isolated from other neighboring populations in the French Vosges, Germany, and Switzerland. The species also faces major threats such as illegal killing and road collision. It is listed as endangered in the IUCN French Red List of threatened species. Because this population was founded by a small number of individuals and remained small and isolated, it is especially vulnerable to reduced level of genetic diversity and higher risk of inbreeding. These processes can further reduce adaptability and increase extinction risk. Therefore, the key question of this work was whether this population retained enough genetic diversity to remain viable in the long term. I am now introducing the Œil de Lynx, Eye of the Lynx project, led by the SFEPM, Chrono Environment, and Gecolab Laboratories. This project aims to update and refine our knowledge of the genetic status of the lynx in the French Jura and Northern Alps. Fieldwork was carried out by a citizen science network of more than 200 volunteers formed and trained by the SFPN. This network was taken to collect Eurasian lynx scats. Without them, this work would simply not exist. Our goals were to first evaluate genetic structure, then to assess genetic diversity and inbreeding, and then to investigate gene flow within the population and with other European populations. The laboratory analyses were initially carried out by Chrono Environment Lab, including 174 lynx cat samples and 18 microsatellite markers. Microsatellites are short repeated DNA regions whose length varies between individuals, which make them useful as genetic fingerprints and thus for what we call genotyping. The analyses were taken over or continued by the Gecolab with 53 additional samples so far and more to come, and by using 20 microsatellite markers from the Lynx European Consortium. The genotyping successes were 62 and 56% respectively, which is actually quite good when analyzing scats. After filtering genotype profiles with too much missing data and identifying recaptures, individual samples several times, we ended up with 74 and 98 uniquely identified individuals respectively. Additionally, the Gecolab determined the sex of these individuals, identifying 31 females and 67 males. We would like to warn you that these figures do not represent the actual population size. They only represent the minimum number of lynx detected over the 6 years of sampling. Some individuals detected early in the period may no longer be alive, and others may simply never have been sampled. Anaïs will now present you the results. You'll see the logo of the labs on each slide, indicating which laboratory contributed to the analysis.
Thank you, Laurie. So let's start with the genetic structure. We use the structure software to test whether the links from one or several genetic groups. The best reported model was K=2 for analysis from both labs. So at first glance, you might think, great, we have two populations, but not exactly. Because when we plot the cluster memberships on the map, something quite striking appears. The two clusters are completely mixed across all departments. They don't correspond to geography, and they don't correspond to sampling year either. So despite this statistical signal, there is no meaningful spatial or temporal structure. So how do we explain this? This pattern is very common in populations that have experienced a strong founder effect followed by demographic expansion. The reintroduced lynx population originated from a small number of founders, which likely reduced its initial genetic diversity and homogenized allele frequencies. As the population expanded and is still expanding today, these founder allele frequencies simply spread across the landscape. So what we observe now is not evidence of multiple distinct populations, but rather the genetic imprint of a small founding group that has grown and dispersed over time. So the lynx population in French Jura and Northern Alps is genetically homogeneous, likely shaped by its reintroduction history. Now let's look at genetic diversity. We found an allelic richness of 3.18 and a positive inbreeding coefficient of nearly 0.20. For large carnivores, these values are low. For example, the allelic richness of the natural population in Scandinavia that has never been reintroduced and has many more lynx is 4 to 6. These values are also lower than in source Carpathian populations. This is fully consistent with a small number of founders and a long-term small effective population size in France. Finally, we assess genetic differentiation first in our study sites between French departments using GST and DJust. You can see that the values are very low, meaning a significant gene flow and a low differentiation. This reinforces previous conclusion, weak genetic differentiation. Then we assessed genetic differentiation with other European populations. We calculated FST values with genotype data from Gajdarov. Pairwise FST revealed a very large differentiation between our studied population and 5 other European populations, including the Carpathian source population. So, in our study site, we see a low differentiation between French departments. This pattern likely results from continuous gene flow or shared post-reintroduction history, or most likely both. But at the European scale, we see a very large differentiation with other populations, including the source population. This differentiation likely reflects both the founder effect and subsequent genetic evolution, possibly intensified by the complete isolation of the population since the reintroduction. So, what does all this mean? The studied population showed no intra-population but exhibit low genetic diversity and risk of inbreeding. Its isolation from other European populations points to a persistent founding effect, which is typical of reintroduced populations founded by few individuals. Similar trends were observed in other Eurasian lynx populations in Europe, such as in the Dinaric Mountains. These results highlight the need to improve large-scale monitoring of genetic health, as 10 of the 11 European lynx populations are typically transboundary, along with increasing connectivity with neighboring populations to ensure sufficient gene flow, while reducing anthropogenic mortality, especially collisions and illegal killing, which are major obstacles to gene flow. And finally, to enhance social acceptance of the species, and thus ensure the success of conservation efforts. Citizen science could serve as a valuable tool. So to wrap up, the Jura and Northern Alps lynx population in France is genetically cohesive, but low in diversity, shaped by founder effects and isolation from other populations. So generally speaking, the reintroduced lynx population has survived but needs help to thrive. So we are reaching the end of our talk. Laurie and I thank you for your attention, and we will be glad to answer your questions.
Thanks, Laurie and Anaïs, for this impressive work and sharing those results with us through, if I may add, a very good-looking presentation. While Q&A is filling up, I have a question of my own. Having seen wildlife management in South Africa while living there, where some populations are relatively closed and genetic diversity is maintained through translocation, cheetah population comes to mind. I was wondering whether France might eventually adopt a similar approach, or is there a focus on improving connectivity and establishing corridors between these populations?
Yeah, thank you, Joan, for your question. We were actually expecting this one. So our work is like part of a national action plan to, among other measures, gathering some scientific data about the necessity of this reinforcement of the population. There was also in 2024 a viability assessment that explored different scenarios of the demographic and genetic evolution of the population by 2130. And they also came at the same conclusion that we need the reinforcement, genetic reinforcement. But this one can only be successful with these additional measures, such as reducing mortality, improving connectivity, and also improving the social acceptance of the species. All these measures to guarantee the success of this reinforcement project.
Great, thanks for that. I see we do have a question. Wu, would you mind unmuting yourself and ask a question?
Yes, Laurie, thank you very much for that talk. There's two questions. One is that probably just a short one for clarity. You had two labs doing work, and they both had different numbers of individuals, this estimated from the genetics. Are those additive, or are they like the same individuals and there's overlap, or could you add them together and that's kind of the total number that you detected? And that second question, I'll wait till you answer that and I'll ask the second question.
So thank you for your answer. Yeah, it can be a little bit confusing, but we didn't use the same microsatellite markers. So these two figures cannot be additive to each other. And so we kind of identify different genetic individuals. The main reason for that is that because we didn't have access to the list of this consortium, a list of microsatellites. So that's why now that the JCOLAB is taking over the analysis, they are using this list that can also give results comparable with other European populations.
Okay, thank you. And the second question was, is there a method for using this type of data to estimate populations?
Yeah, that's really a great question. The problem is, we used a citizen science network, which has a lot of advantages, such as like the area of prospection, the number of samples. But the major drawback is the heterogeneity in the spatial coverage and also the time. Like, for example, we have no idea how much the collectors, like the size of the area prospected, nor the time. So it was difficult to use, for example, spatial recapture modeling for estimating the density because we couldn't model the sampling effort.
Okay, thank you.
Thank you again, Laurie and Anaïs. It was great presentation again.
Thank you.
Next, I'm excited about our next talk. After hearing about cat ecology and conservation challenges, we now gonna shift to the topic of data itself, which is such a fundamental part of any research project. I'm sure this topic will resonate with many of you. So with that, I'll introduce you to Jason Holmberg, Jason is the director of the Wild Me Lab at the nonprofit Conservation X Lab, which builds AI-enabled infrastructures for research and conservation. Jason's work focuses on multi-species machine learning for mark recapture population studies, scalable forest monitoring systems, and collaborative data platform that supports conservation science across terrestrial and marine ecosystem. Jason, thank you for being here and welcome. The floor is yours.
Thank you very much. How's my audio?
We can hear you perfectly.
All right, and then let's check the video. Is the sharing okay?
It's still coming up. Yeah, perfect.
All right. Thank you very much to Panthera and this community for welcoming me in. I'm a little bit of an outsider in that my career is in tech, so I'm more on the data management and artificial intelligence side of conservation. And my job is to help researchers like yourself use data and leverage AI to accelerate your research and hopefully on-site conservation action. I want to clear up some terminology first. Conservation X Labs is the nonprofit that I work for, and you may have heard from a previous presenter from our Central Asia program. We also have programs such as, let me advance the slide, the Sentinel program, which is an AI-enabled camera trap. The device allows you to use AI and instant notifications on the edge and plugs into existing camera traps so that you know when an animal crosses in front of your path and which species it is. That is one of our separate programs. And the program I represent is a distinct unit inside Conservation X Labs called the Wild Me Lab. So you may hear us interchangeably called Wild Me. We used to be our own nonprofit and merged in, or Conservation X Labs. If you think about the Wild Me Lab as an AI startup in a nonprofit shell, we have front-end and back-end software engineers, machine learning engineers, and even full-time customer support. And by customer, I mean supporting users such as yourselves who are logging into largely our Wildbook platform. And currently we support over 2,500 users tracking 319,000 individual animals by photographs and their individual identity as it appears in those photographs across about 1.6 million photos. So in the Wild Me Lab, we're leveraging AI and good data management to help multiple researchers like yourselves for marine and terrestrial species track their populations, come up with better population estimates, and ultimately try to generate better on-site conservation action plans. An important aspect about our lab is we have no ecologists on staff. We don't claim to understand the ecology or to write papers. We're non-competing. We are the technologists trying to build tools in support of your work. And specifically, these tools are looking at a couple of specific areas, such as the long gaps between population assessments. Sometimes it's 5, 10, 15 years between good local population assessments. There's a slow iteration cycles of protection strategies because they're dependent on good data. There's a gap between promising technology like AI, which is often implemented as complex Python, and then successful scalable field applications that actually work in the field. There are funding gaps that make it difficult to fund AI projects, skills gaps, and experiment versus engineering experiments where sometimes an academic experiment that's published in a paper doesn't translate into something that's usable in the field. And since 2002, we've been leveraging our distinct engineering skills to help solve conservation problems in the field. We do this largely in our Wildbook program using AI and really two steps of AI. One is detection. So this is whether it's an African wild dog or a humpback whale in the bottom right. We use AI to draw bounding boxes around the features of interest. And that AI that we call detection is actually multiple AI steps, such as one, localization, which is drawing the bounding box around one or more animals in the image. Two is viewpoint prediction. Are we looking at the left or the right side of the animal, which is important because they have different patterning often on the left and right. We have something called an orientation network, which can help predict how rotated is this animal in the image, and can we snap it to a horizontal perspective so that comparing individuals is done at common perspectives. And we have additional AI steps that we really bundle into a step we call detection, and it's mostly about finding the animals in images. Next, we have re-identification. So if an animal based on its physical structure, either obvious natural patterning or structure such as fin shape, fluke shape, face structure, can be re-identified reliably by human researchers who can also validate the AI predictions, which is important, then we can deploy AI to help speed and scale re-identification of individuals, allowing you to upload images, have the detection phase, draw bounding boxes, predict the viewpoint, and then the re-ID phase's job is to take those bounding boxes, search through a catalog, and give you a list of its top predictions of who am I looking at. Here you can see examples of obvious visual patterning matched across time and different images. Now, small wildcats are interesting, and in 2025, in our whiskerbook.org platform, we added support for all of the small wildcat species. There's an important caveat here, which is for many of these species, they're data deficient and there isn't training data for AI, nor is there an understanding of how well the AI performs on them until we have at least a basic validation data set. However, the world of AI has advanced into what we call these foundation models, whereby if we have enough data for other species, especially similar species, then we can do something that's called zero-shot learning, which is, can we translate what we've learned from ocelots, Eurasian lynx, Iberian lynx, leopards, jaguars, snow leopards, and create a detector that for other species, hypothetically small cat species, does a great job of drawing bounding boxes around those animals, even if the AI training data has never seen that species or examples of those individuals. So in our AI lab, we created a model that you might be familiar. It's comparable to Megadetector, if you're familiar with that for camera traps, but it's trained on our community source data as well as some public data sets. And its job across marine and terrestrial species, including some of the species I just listed, is to draw a bounding box. And with zero-shot learning, we can cross-apply that to small wildcat species and allow us to draw bounding boxes and even predict viewpoint left versus right on small wildcat species that we've never seen. So we're cross-applying what we learned from other big cats and other species to small wildcats. For AI and re-ID of individuals, we suffer from the same problem. There isn't a priori a great data set of servals, for example, and serval re-ID. So how do we serve that community with re-ID, even if the AI models have never seen that? There again, we leverage zero-shot learning. Our lab creates the MUID v4 deep learning model, which is trained on 90 species. So oddly enough, ocelots, Eurasian lynx, Iberian lynx, leopards, and others are represented from marker capture catalogs in the AI training data. However, they're also included with giraffe, leafy and weedy sea dragons, humpback whales, and a variety of other unusual species. What MUID v4 has shown is that if we increasingly incorporate visual marker capture catalogs from many species, we can serve all species better with slight improvements in accuracy due to the fact that we're just leveraging larger amounts of training data and we're allowing the AI to get this superhuman sense of who am I looking at, and to focus less on things like background matching, which camera trap re-ID suffers from, and more on shape, structure, and visual patterning of each species. So the hypothesis here is that this translates very well based on what it's learned from morphologically similar species, but also very diverse and different looking species, and it cross-applies to small wildcat species. As we list here, we do have some small wildcat training data in the model, which it benefits from, but for many species, we're doing zero-shot learning. We also leverage an older computer vision algorithm called Hotspotter, which is not deep learning. It is simple, pure brute force texture matching, which has its own challenges and benefits, but it doesn't even understand what an Iberian lynx is or a jaguar. It just looks for corresponding textural patterns in potentially matched images. You can see a quick list in our current iteration, which just came out in January, of the MUID v4 model, our fourth training of this big foundation model, how we do with mean average precision and top one. Mean average precision is if I only have one other example photograph in my catalog, how well do I do at creating the correct ID prediction? Top one is if I have this individual represented one or more times in my ID catalog, and I've got up to 3, 5, 7 chances to make that ID, how well do I do in predicting it in the top one rank? So mean average precision is generally a little bit lower because that's your worst case scenario. I only have one chance to get the correct decision right. Top 1 gives you a few chances, and then you've got top 5 and top 10. And ultimately, what we're trying to do is give you a ranked list and say, here's the top 10 individuals we think are correct, maybe assign even a confidence level to that in the future, but give you the ability to search through large catalogs quickly, see predictions of individuality, and accept those predictions as part of the curation. We also have a new algorithm in development that's in quality testing right now. It's called MUID in-depth. And we actually run the same MUID decision, and then we use a second pass algorithm to take a second look and say, all right, we've made these top 50 predictions. Now look at the fine-grained features, the evidence of patterning on the animal, and re-rank these decisions and make a better prediction. And what we're seeing is for small wildcat species, a pretty big benefit in using this second re-ranking system. We bundle all of this AI in the convenience of your browser inside what's called the open source Wildbook platform. And here, one of the Wildbook platforms is called whiskerbook.org, which allows you to get an account, log in, upload photographs, run the detection and re-ID stages right from your browser. No GPU needed, no advanced Python knowledge needed. This is an example of an encounter record that's been submitted. AI has already drawn the bounding box. My next step after reviewing this data for date and location inside the Whiskerbook platform would be to click New Match and ask it to examine the catalog for this specific area and say, give me a list of potential matches. From here, I could assign a new ID and say this is a new individual, it's unmatched, or assign an existing ID and create larger and larger records of individuality. We also have full export formats, including exporting email addresses for your data contributors, exporting a Google Earth file, exporting Excel files, GIS shape files, and then files for population modeling applications like Program Mark. This platform is used across the globe for a variety of wild species. We have a Sharkbook platform, Flukebook platform, but for wildcats, importantly, we're focusing on the Whiskerbook platform where we added the support. It's researcher focused, it's nonprofit managed, professionally managed, and it has this concept of silo security. So you don't have to share your data with other researchers, but should you optionally want to, you can extend invitations inside the Whiskerbook platform to allow others, whether they're part of your lab or your program or from other land areas that are connected, to share your data and begin creating these larger and larger catalogs across boundaries, time, projects, etc. I believe that was my last slide.
You caught me off guard here, Jason. Great, thank you very much for this very interesting talk. A lot of exciting points here. It's interesting to see Hotspotter is still currently used. But yeah, thank you very much. I'm gonna leave people type their questions. But while they do that, it's nice to see you highlighting how data integration and AI can open new possibilities for monitoring and studying small cats population. So thank you for that, for this technology touch to our symposium. Maybe I can ask, what are the biggest challenges when implementing AI-based identification system in wildlife research?
Scaling can be an issue. So for example, we just had a record where in our Wildbook platform in a single day, researchers across the globe ran 9,900 re-ID jobs, meaning that they asked 9,900 times in one day, who am I looking at? And so we start with small data and serving small projects, and we keep increasing the volume of data that we're managing and the AI, and we have to keep scaling our backend to make sure that we meet the demand. So scaling is a challenge, but we very much enjoy it because there's a lot of efficiency in being able to add new species very quickly by building these big models, by running in a centralized platform so that we don't have to replicate many small projects and sort of divide our time among them.
Great, thanks. I was wondering another question regarding data ownership when collaborators are sharing those data on your platforms? Do they keep total ownership? Is there timelines on those? How do you work? Do you reuse those for your catalogs? Is there agreements on those?
So because we're engineering, we can sign almost any MOU. We're not on site. We're not competing in any way. We do not share data without your express permission. By putting data into the Wildbook platform, you continue to own the data, but you do give us rights to train AI for the benefit of this community. So we don't turn around and sell those AI models or do any of that. We build the AI models, deploy them right back to the Wildbook community. So you do not give us ownership of your data when you submit data to the Wildbook platform.
Great, thanks. We have a very good question from Blair. Blair, do you mind unmuting yourself and ask a question?
Can you hear me all right? And sorted out my technology issues from earlier.
Yeah, we can hear you.
Excellent, thanks. Super interesting work, Jason. I can imagine that you are going to get swamped with a lot more inputs following your presentation here. I am curious, given the nature of wildcats and how hard they are to photograph, what kind of image quality do you need to actually be usefully continuing to train the AI for something like wildcats? Does it have DPI minimums or file size minimums or anything like that that potential users should be aware of? Thank you.
Excellent question. One of the dirty little secrets of computer vision is that we downsample almost everything to about 400 by 400 pixels. So the great answer to that is we can take even small images. Of course, big high resolution photos are amazing, but under the covers, they all get downsampled. And so generally resolution isn't much of a problem as long as the feature that we're matching, whether it's face structure or patterning, is clearly visible. Even with some blur, we've even seen like the tail of a leopard for like the last 6 inches matched with something like Hotspotter that just does brute force pixel analysis. It's pretty impressive. In the training data, when we begin training the big model, we have to be a little selective because we want the AI to be able to at least see the feature. But in real world application, visual diversity of different presentation angle, different lighting conditions helps minimize the probability that we learn background matching and instead helps focus on real world re-identification across quality. Even today, I was just made aware of a paper of ways of simulating some of that diversity by quite frankly, introducing data quality problems to help generalize the model across real world data. So while big, beautiful images are great, we actually downsample most of them and we want broad visual diversity. So even your bad images may end up getting trained in.
Amazing. For a lousy photographer like myself, that is music to my ears. So thank you. Terrific stuff.
Thank you.
Thank you, Blair, for the questions. Rue, I'm so sorry we're running out of time, but please do reach out to Jason. Or Jason, if you may have a moment to answer the question on the QA tab, that would be amazing. Thank you very much again for your presentation, Jason. We're going to jump on a quick 5-minute break, and I'll see you shortly after. Okay. Welcome back, everyone. We are now going to jump into our very last 3 speed talk of the symposium. And we're going to start first with Emily Madsen. She is a PhD researcher at the University of Oxford's Wild Crew and is presenting on behalf of her co-leads Divya Rana and Carol Sator and the wider team. Emily's background is in carnivore research in East Africa, while Divya and Carol are specialized in Asia and South America. Emily, welcome and thank you.
Hi. Can you hear me okay?
Yeah, perfectly.
Perfect. And are we seeing everything?
Perfectly too. Full screen.
Great. So we gave ourselves the difficult challenge of trying to synthesize all of the research that you have all been doing. So we conducted a very large literature review in multiple different languages across multiple different platforms in not just the Latin alphabet languages, but in other languages too, such as Chinese and Russian. And our global search terms included the Latin genus names when we were searching the Latin alphabet, but in other non-Latin based languages, we included local names as well. Just to note, we excluded the domestic cat, and we collated in all the research published papers from 2000 to 2025. We were very surprised by the number of results we got, a bit like the submissions for this symposium. So to start off with, we've got 10,213 unique results. We then filtered these through a number of different inclusion and exclusion criteria, and our final group was 4,598 papers that we then subsequently extracted data from. And we're going to whiz through some very preliminary results. So the first one is the geographic distribution. As you can see, Europe and Central Asia were the biggest contributor of this type of small research on small cats. But when we look at it by country, then the USA is by far dominant. When we look at it by species, you can see that there's a huge range of number of studies across the species, with most of the most commonly studied species being least concern, and our endangered species actually receiving the least amount of research. So we have our Eurasian lynx at the top there, and then the Chinese mountain cat right down at the bottom. This is just referencing back to Axel's slide on the first day. These are the only two species I think have not been covered by any talks in this symposium. So it's the rusty spotted cat and the Chinese mountain cat again. We also extracted information on data sources and research objectives. So just a few highlights here. You can see that in North America and in Europe and Central Asia, they were very dominant, much more dominant than other regions on GPS genetics type studies, probably referencing the amount of funding that's available. And then when we look at research objectives, it is population ecology type studies, which are the dominant across all regions. We then ran some GLMs looking at the drivers of research effort by species, by country, and by region. And a few highlights here. So by species, IUCN status is the strongest predictor. And then an interesting find we thought is that an increased percentage of overlap with other small cats has a negative effect on the amount of research on a small cat. So it seems to have, if you overlap with a lot of other small cats, there's some sort of dilution effect. Then when we look at by country, unsurprisingly, probably GDP is the strongest predictor. And then by region is the number of biomes. So the diversity, the more diverse a region is, the more research there is being conducted there on small cats. Then finally, we also did some author network analysis. So this is of the 500 most common authors within all of these papers. And here it was Europe and Central Asia was the region that was the most dominant, with over 50% of the authors from that region in that top 500, followed by Latin America. And that is a very whirlwind run through. The paper will hopefully be out soon, so watch this space.
Great, thank you, Emily. Wow, what a work! I'm sure. While people are typing their question, yeah, thank you very much for this very informative presentation. Also, well done for summarizing this work in such a short talk. We really appreciate. I don't see questions yet. Maybe I'll have one. Have you noticed any shift in research focus or priorities over this amazing review that you guys did?
Yeah, so we restricted the review from 2000 to 2025, partially because we didn't want to, there's a lot of differences between regions before 2000 about whether things have been digitalized as well and things. So we wanted to keep as minimal regional bias as possible. Over temporally, we didn't see any clear patterns. Obviously, there's been the biggest pattern we saw was that there's a huge increase in camera traps being used, and that's having an effect on what type of studies we're seeing coming out about small cats, because it's a lot of this bycatch type of study. But yeah, that wasn't. I think we've got a lot of that. We discussed that in the paper a bit, but it's not super surprising, really. We see that in a lot of this type of research.
Looking forward for the paper. Thanks, Emily. I see a question from Wu. Wu, if you would like to unmute yourself.
Emily, yeah, it was really interesting that it's a throwaway thing actually. It was that I noticed you're negative with negative effect of number of other cat species in the area, and from a Scottish perspective, we have one cat and we are so focused on it, and I think that's probably a big driver. If you didn't have many cats in the first place, cat species, you get really interested in the ones you've got and very concerned about them if the population's declining as well. But I don't know what you think of that. Maybe.
No, I think you're right. I think if you've got a lot, then you might, there's a lot less focus that's going to be paid on in the individuals. I think in Tyler's talk earlier, he mentioned how there's been a huge interest suddenly in the ocelot in Trinidad, and it's because it's their one small cat. It's very, very exciting to everyone. So I think, yeah, it's quite a common pattern to see it that way.
Yeah.
Thanks. We have another question. I'll be the last one before we continue, from Victoria. Victoria, would you mind unmuting yourself and ask your question?
Yes, thank you, Jolene. Congrats, Emily, for the research. I would like to know, please, is like the authors and the regions, they are all connected, or like the numbers of papers from the US were all made by local researchers, or they use data from around the world, like like this?
So that's so thank you. I missed that out. I meant to say it. So the regional stuff, when it came to where the papers are from, was based on where the research happened. So it's probably, if we looked at sorry, something happening. If we looked at by where the authors are from, we'd see even more of a regional bias, because I imagine a lot of the research happening in some countries is actually from researchers outside of that country. But that was really difficult to identify from the papers themselves. We'd be having to make assumptions that we weren't comfortable making based on where people's research institution was. Doesn't necessarily mean that's where they're from and things like that. So we focused instead on where the study was happening. And then the author network, obviously we did use their institution.
Great, thank you very much, Emily. Thanks, Victoria, for the question. We're gonna continue. Next, I'm again excited for the next talk. I feel I have been saying this all day, but what can I say? He is presenting on two passion, mountains and felids. So next we have Claudio Sillero. Claudio is a conservation biologist at the University of Oxford. His work spans ecology and conservation of threatened species, with a particular interest on carnivores and mountain ecosystems. Here you are, you and me, Claudio. Thank you very much and welcome. Please take it away.
Servals are widespread in sub-Saharan Africa. They tend to be associated with long grass and wetlands and so on. But primarily, what determines the presence is a high density of small mammals, because they are the small mammal specialists. There's very little known about them at high elevations, and we felt that we could use opportunistic field observations to have a better understanding of them in the Ethiopian highlands, and we went beyond reporting known altitudinal limits. So that's great. 90% of the land above 4,000 meters in the continent is found in Ethiopia. That's why I like to refer to these highlands as the roof of Africa. And there are islands in this landscape, which are these huge mountain massifs where Ethiopian wolves reign supreme as apex predators. And those are the areas where we work. And when you look at some of these highlands and moorlands, you might think that they're quite poor and unproductive, but in fact it couldn't be any farther from that. There's a very high density of small mammals in these places, which makes them incredibly appealing to carnivores and also to birds of prey. This is my favorite, the giant moor rat or root rat, which is the preferred prey for Ethiopian wolves and for many other mid-sized carnivores. And we can see in the physiognomy of Ethiopian wolves how prepare and specialize to this life. They are very long limbs, large ears, and prolonged muscle. But you could see how they also have a feline counterpart in the servals, which have equally long limbs and round, large ears. So great small mammal predators. And they're part of a carnivore guild that's being pushed high up into the Ethiopian highlands. We see very steep slopes here that lead on to the Afro-Alpine proper. And we could see agricultural processes that keep pushing some of these systems ever higher up the mountains. And we are now witnessing this high diversity of carnivores in sort of mountain refuges. And Ethiopian wolves as several are quite present there. So we use information that is collected by the Ethiopian Wolf Conservation Program monitoring teams opportunistically to look at where these servals might be. And we had 311 independent observations over 30 years across 6 mountain ranges. It doesn't seem a lot, but it took quite a while and effort to collect. And they reflect these tall grass, high altitude swamps. They seem to be prevalent on wetland edges and on the base of low rocky cliffs. And among some of these observations, we also have a handful of wolf-serval interactions that I will refer to. And in the map, we can see at the bottom right the Valley Mountains, which is the largest of these 6 ranges. So first, let's look at the altitudinal range. And we knew that servals like mountains, and the highest previous record in publication is in Kilimanjaro, 3,850 meters. And we found a range of 3,000 to 4,300 meters. And we don't have data below 3,000 meters simply because we don't work there. 40 of these sightings were above the current record, as it were. And the highest, at 4,300 meters, is in Wahit, which is Ethiopia's second highest mountain. Among these records, we also have a few melanistic animals. So they're not uncommon in mountain forests, glades farther down the mountain, but we also found a handful up in the Afro-Alpine proper, up to 3,500 meters. Looking at the profile in terms of daily activity of these sightings, we can see that mostly diurnal observations displaying a bimodal pattern, and this seems to indicate as well the availability of a small mammal prey. Also a slight bias because we don't work as hard at night as we do during day hours. The sightings are largely of single animals with a handful as pairs, and we have one sighting of a family with kittens that would have been born in June during the wet season. Some incidental captures as well, which place them within the weight range, so there is no obvious displacement on weight and so on. And coming into the favorite aspect of this observation is what's happened when they encounter wolves. I mean, these two seem to be ignoring each other, although you can see the bristles rising in the serval. So a grand total of 10 observations. The one in the picture only from 4 days ago. The aggressive interactions tend to go 2 ways. I mean, if a serval meets a wolf on their own, the wolf is most likely to take off and run away. Whereas if a serval meets a wolf pack or a wolf group, it's more likely the serval that makes a run. And we had 2 non-aggressive interactions, as it were, where a serval quite contently sat right next to a wolf pack. So it takes all kinds. So basically, we can place these servals into those Afro-Alpine refuges, and we just wonder whether they're safe there or not. We can see intra-guild competitions being an aspect of concern, particularly with this ever-growing pressure of agriculture up the slopes. There is a high pressure on wetlands as well, and those are getting increasingly fragmented, so that might have an impact on the use of microhabitats. In some areas, particularly in the north, there is some retaliation due to livestock predation, and sometimes they are unintended target because some other carnivore species might have been the culprits. Disease is very much in the system that's been central to our work with the wolves and might affect the servals as well. And compounding effect of all these might result in eventually in low genetic diversity. So we still have the servals there. We need to learn more about them. They're quite present right across the highlands, but they're facing serious concerns like most other carnivores. Thank you.
Thank you, Claudio. Amazing talk. Thank you very much for this excellent speed talk. While people are posting their question in the tab, I can't help to draw a connection with the idea of mountain becoming a refuge for wildlife in an increasing human-dominated landscape. Do you see that more specifically with a species per se, or is it the general system?
Well, there are certainly processes that have an attitudinal axis. I mean, people displace themselves up and down the mountains. They might move towards new grazing land. They might move away from cold weather and misery into urban areas. So there is that constant. There is pressure for livestock that gets pushed out from agricultural land up into pasture. And animals seem to respond to that. Fire also is of relevance because there seem to be different dynamics, and sometimes with increasing pressure, the fire increase, and that might generate new habitat for some species, but might be bad for others. So clearly there are dynamics that we need to understand better. But what is clear is that mountains continue to be a refuge of biodiversity. They are very high in endemism, and they are incredibly important reservoirs for water and other natural resources. So I think bringing a focus into the importance of protecting mountain systems is quite important, not just for biodiversity, but for humans as well.
I can't agree more. Thank you, Claudio. We do have a question in the Q&A from Bilal. Would you mind unmuting yourself and ask a question?
Hello, Claudio.
Hello, Bilal.
It's a pleasure to hear after so much time. Thank you so much for such a nice presentation, such an informative one. I just have a question that is there any interaction any presence in the local folklore about serval and Ethiopian wolf interactions in particular?
No, they seem to feature in the sense that you occasionally see skins being used by people. There might be some use for medicine in some areas, but in terms of actual tales that bring the two together, nothing that I'm aware of. I mean, the wolves do feature, but nothing that brings both serval and wolves together. But now you planted that idea, so I might have to look into it. Thank you.
Thank you so much, Claudio. Thank you so much.
Thank you, Bello. Thank you, Claudio, for this excellent presentation again.
Thank you, Shalini.
For our next and last presentation, it's my pleasure to introduce someone I deeply respect as both a person and a researcher. We have Hugh Robinson. A little anecdote here, actually. We have asked every presenter a little intro to help us introduce everyone accurately. And I received a very modest one from Hugh, but I'll stick to it. Hugh is the director of Panthera's Applied Science Program and is based in Montana, Missoula. Hugh, thank you for your time and thank you for being here. The floor is yours.
Nope, it's not right either.
You need it there, Hugh. You just need to go on top there in the middle. Yeah, and swap.
Okay, but I can't see my notes, but that's all right. I'll just wing it. We recently added co-author a second study area to this to our study. So they're not listed here, but we will add them as we move forward. Camera trapping has become ubiquitous in conservation science. There we go. In wildlife research and conservation, this figure from Blount et al. Provides an illustration of the increased use of cameras in recent decades. While some of these surveys are designed to monitor multiple species or communities, most often there's a single species of interest, and yet cameras are non-selective, so collect images of anything that passes through the viewshed. We were interested in finding a way to tap into this bycatch data. Specifically, we were interested in its use for monitoring small cats and using existing data sets as a means to identify populations in need of attention. Informally, that's something we referred to as an early warning system. A model type we thought might be useful are dynamic N occupancy models, first described by Rossman et al. In 2016. These models are a combination of N mixture and dynamic occupancy models. They use detection, non-detection data rather than count data and account for imperfect detection. Farr et al. 2022 expanded on the original model by incorporating multiple species in a community model. We used bycatch data from two study areas, Kenyer in Malaysia and Welgevonden in South Africa. In Kenyer, the focal species was tigers, but they also captured photos of a number of small cats, including clouded leopards and leopards. In Welgevonden, the target species was leopards, while bycatch data were African wildcats and servals. Our methods consisted of two basic components, simulation and comparison to estimated densities and an associated population trend. In the original Rossman paper, they tested the model on data from a 25-year continuous time series. We used simulation to test how the model performed with less data, as well as how imposing a post hoc sample grid affected our estimates. For comparison to SECR estimates, we used the Pearson correlation test, taking the SECR estimates as truth. Our simulation suggests that the model is robust to data gaps and shorter time series data and may perform better when a sample grid cell size is larger than your species of interest's home range. When compared to SECR trends, the model performed well for 3 of 4 species, clouded leopards being the exception. Finally, in an application to bycatch data, after establishing that the method might be good, we applied the model to 2 species from South Africa and estimated these trends based on data originally collected to monitor leopard density. So the use of dynamic N occupancy seems promising and may also have application to prey species or any other bycatch data. We have the clouded leopard puzzle to figure out, and we'll be submitting a manuscript soon. Here's a picture of a cougar for you. Thanks.
Thanks you for finishing your talk on a big cat species. Thank you very much. I'll let a couple of minutes for people to jump on the Q&A, but thanks for presenting such interesting findings. I'm obviously biased, I would say, but I think, yeah, this is valuable work that could bring significant impact, I think, for the field. You mentioned the clouded leopard estimates. Why would you think they don't seem to agree here?
Can I go back and share my screen again?
Angus, I think you need to give him.
That's okay.
Yes, no, you should still be able to, yes.
Okay. I'm just going back to... Actually, after the start of this presentation, I don't know why I'm...
So good.
Never mind. The clouded leopard issue is interesting. With the data from Kenya, they only had 3 annual estimates or 3 estimates over 14 years, I think. And whereas with the N occupancy, we are looking at an estimate for every year, which is creating a kind of a conflict between our estimates and theirs. In the end, it's actually interesting that over the span of the entire time series, the two methods do agree. Both are suggesting a slight decline in the population. It's just that correlation, the Pearson correlation method is maybe a little finicky about like all the data in between as opposed to the start and end point. So it'll be interesting to explore that further and try and figure out exactly what has caused the difference.
Thanks, Hugh. Thanks for this extra info. I have a few questions on the Q&A tab. Maro, would you mind unmuting yourself and ask your question?
Hello, Hugh. Can you hear me?
Yep.
Thank you very much. Very interesting. I was wondering if you were able to detect an effect of sampling effort, both in terms of number of sampling sites and length, duration of a sampling effort.
One of the benefits of adding the second study area in Kenya was that the sampling that was done in South Africa for leopards is very systematic, and the cameras, the same number of cameras and also paired cameras set up for SECR estimation. The paired cameras are set up in the same location every year, the same number of cameras. Whereas in Kenya, in the Tiger Reserve, there was a vastly different effort across time. And so some years there were very few cameras put up. They were just put up, well, especially during COVID, they were just put up to maintain a certain level of presence in the study area. And so you seem to be able to get the model is robust enough that it can get around those different sampling pressures or levels over years. And again, even with the simulation showed us that you can actually even have entire missing gap years and still come up with a fairly accurate estimate. So I don't know if there's a minimum number of detections or that you would need. We haven't actually gotten into that, but it seems fairly robust to changes or variation in sampling effort and detections.
That's great. Thank you very much.
Great. Thank you, Maro, for your question. And thanks you again for your presentation. Michelle, I have another question. Hugh, I don't know if you can answer maybe on the Q&A tab as we running behind. But thank you everyone again. With that, I'll hand over to Ming to lead the panel with this wonderful group of speakers coming up. Thanks everyone for keeping up with my French accent all afternoon, and looking forward for this panel.
Thanks, Jolene. Thanks for the crossover. Well done today, and it's been a great day of presentations. I really learned a lot. So while we just kind of transition over here, good afternoon, everyone. Good evening in some cases. This is coming up to the final stages of the symposium. It's been a long 4 days, so just want to say thank you so much for kind of hanging in there to participate in this last panel. I hope you guys will find it interesting and kind of relevant to a lot of your work. So without any further ado, I'm going to kind of crack on here. Hopefully, the panel can get their cams on. Right, so while we get everyone on, I'm going to move on. So small wildcats are among the least studied carnivores in the world, yet many are facing rapidly intensifying pressures from habitat loss, habitat degradation, direct killing, agricultural expansion, and human-wildlife conflicts. But despite decades of research, we still struggle to answer some of the very basic questions about many of these species, including how many are there, where do they persist, and whether populations are increasing or declining. One of the central challenges is that, as we all know, small cats are simply very difficult to study in the wild. They occur at low densities, many are nocturnal and highly cryptic, and they often occupy dense rainforests or wetland systems where detection is inherently difficult. So for this panel, we'll focus on the core monitoring challenges and uncertainties that define small cat conservation today. And then we're going to explore how different tools, albeit alone or in combination, might realistically help address these challenges. And so this panel will be somewhat thematic. And as we continue, I am very pleased to introduce the panelists for this discussion. First up, we have Dr. Andrew Hearn. We've heard from him before with a wonderful presentation on day 2. Andy is a carnivore specialist and founding director of the Bornean Carnivore Program, as well as postdoctoral researcher at the University of Oxford. Over the last 2 decades, he has led numerous studies on Southeast Asian carnivores, particularly clouded leopards and other small wildcats, helping guide conservation efforts across Borneo and the wider region. Next up, we have Dr. Sara Williams. Sara is a quantitative wildlife ecologist and deputy director of Panthera's Applied Science Group. Her work focuses on demographic and species distribution modeling, particularly using camera trap data and advanced analytical approaches to better understand population dynamics and species distributions. And finally, we have Dr. Byron Weckworth. Byron is the Chief Conservation and Advocacy Officer at the Mountain Lion Foundation. His work bridges science and policy, focusing on biodiversity conservation and landscape connectivity. He brings particular expertise in conservation genetics and in designing monitoring programs that guide real-world conservation decisions. Thank you all for joining this panel. I'm super excited to have this high level of expertise discussing some, I think, pressing topics when it comes to small cat conservation. So welcome all.
Thanks, Ming. It's great to be here.
Thanks, Ming.
Yeah, thanks, Ming.
Great. So the title of this panel is called From Detections to Density, Rethinking How We Measure Small Cat Recovery. And so kind of with one of our first themes is, what are the biggest uncertainties and barriers to monitoring small cats? One of the recurring questions in small cat research is whether these species, are they truly rare, or whether our methods are simply kind of failing to detect them effectively in the wild. So I kind of want to pitch the first question to Andy. Andy, who is someone who's arguably studying some of the world's rarest cats out there, the flat-headed cat and bay cat. I was just going to get your thoughts on, are low detection rates primarily an ecological reality, or are they partly a methodological issue? What are we looking at here?
Yeah, I think there are a number of factors shared by many small cats, almost a perfect storm in a way, that shape and the challenge of monitoring these small cats. Low detectability linked to their cryptic ecology, as you mentioned, the mismatch between the monitoring methods that we typically use and small cat behavior, the difficulty of identifying individuals. Some, of course, are spotted and you can identify them. And then they're often patchy distributions linked to specialized habitats. And I think importantly, it's the interaction between these kind of factors, this perfect storm, that gives rise to many of the monitoring difficulties we face. Many small cats occur at naturally low densities, maybe not. They're nocturnal, they're secretive, and they're often occupy structurally complex habitats such as dense understories and wetlands. And all of this leads to very low detection probabilities. But as you say, is that low detection probability actually due to rarity? And I think on some occasions it probably is, and on others it may not be. Let me give you an example from Borneo, because that's where I've got my speciality. We've got the marble cat and we've got the clouded leopard. Now, typically, and this is found throughout studies from north to south, we find that the marble cat is detected very infrequently, whereas the Sunda clouded leopard, again, a bit of a ghost cat, but much more able to detect them, much higher number of captures per unit effort. But actually, if you drill down and you're able to get enough data, and we've been able to do this with a few study sites to actually run some density estimates using SECR. We find that marble cats are, and again, our density estimates are not necessarily that robust, but our estimates are suggesting that the cats are kind of bouncing around the 7 to 20 individuals per 100 square kilometers, whereas clouded leopards, the ones that we record frequently, are only about 1 to 3 individuals. So there's an example where actually it's not necessarily just, you know, the detection rate that we can look at, and that can be due to a number of things.
Oh, wow, that's incredible. I mean, camera tracking marble cats is notoriously difficult. I can't even remember when I've got a clear image of one, particularly if you're using their pellets to ID individuals for you know, the spatial capture recapture analyses for density. Yeah, that is very interesting. And so, I mean, Sara, from an analytical point of view, like what do you need from the field in order to kind of overcome some of these kind of uncertainties and barriers to wildlife monitoring of small cats?
Yeah, it's kind of the golden question that I deal with all the time. And in the work we've been doing with the dynamic and occupancy models and a lot of the data that we've got through Panthera, and it's come up several times in this conference, we're talking a lot about bycatch data. And I think one of the trickiest methodological issues is that when people are setting up projects for specific larger cat species, they're setting up cameras in very particular ways to get the best images that they can to identify those individuals. And so when we're using bycatch data from those very purposefully designed and very purposefully set up camera trap studies, these cameras are not set up potentially very well to capture the small cats. You know, maybe the cameras are too high. They're a topic that we've talked about, potentially why that clouded leopard estimate doesn't match as well from our occupancy models is that, you know, the cameras are set up in a way that they're not capturing more arboreal small cat species. So I think there's kind of this fundamental challenge in using bycatch data for small cat species and trying to analyze and see, okay, are we really not getting many detections because the cats aren't using the same habitats, they're not coming in the same areas, they're not moving around as much, or is it that our methods are just not capturing them as well? And I think from kind of an analytical point of view, most of our models are made up of two pieces. We really have the part of the model that's trying to estimate the ecological truth, what's really going on the ground. And then we have the part of our model that's kind of the statistical part of the model that's dealing with imperfect detection or other kind of, you know, portions of the study that we're just accounting for. And so I think using data simulation is a really good way to look at, okay, we're going to make up what truth is. We can set it up based on our best knowledge of the species, of the small cat species that we're working on. We can set up our ecological truth via data simulation and say, how bad can we mess up detection for it to really impact our estimation of ecological truth? And so I think, you know, having kind of these baselines, having this foundational knowledge of a little bit of how does the species move, what habitats is the species using, we can really use data simulation to kind of tease apart some issues related to, are the species truly rare, or are we just not detecting them as much because of our sampling methods?
Right, absolutely, yeah. I mean, when it comes to sampling methods, you're mentioning even the height of the camera can make a difference.
Exactly.
And you know, when we're talking about bycatch, it's such a powerful tool because there's a myriad of data out there. You know, we could be working with other organizations that don't even do anything with cats. They might be working with completely different taxa, but still collecting this data on you know, species that we might be interested in. But even kind of augmenting some of the big cat surveys by filling in those gaps, right? Those camera grids are generally kind of set to kind of 3 kilometers, maybe in 4 kilometers. But in certain areas where they could possibly kind of augment those surveys and kind of, you know, fill the gaps, as it were, and put those cameras in between and kind of get those data on small cat could be you know, particularly interesting. So I kind of sorry. Sarah.
Oh no, I was done. I was just agreeing with you.
All right. Well, I mean, this kind of like leads on to the next kind of theme of the panel, in such that kind of uncertainties ultimately leads to kind of knowledge gaps. And so, what are the critical knowledge gaps? Even though these monitoring challenges are widespread, they don't necessarily affect all species or habitats equally. There are some species that are a little bit easier and some habitats a little bit easier to work in than others. And so the next question, and this is to all of you, would be kind of, where do you see the most significant gaps in our current understanding of small cats?
I can say a first one that just has come up in work that I've been a part of in Panthera the last few years. We were working on, a group of us in Panthera were working on collecting a whole bunch of bycatch data from Panthera surveys, as well as a whole bunch of collaborators that people working in the region had created this huge network of, fellow researchers. So we were working on a range-wide occupancy model for jaguarundi. And while there was like a huge breadth of data, spanning countries from Mexico down through South America, one of these kind of gaps that was strained, that I found kind of surprising at first, was this big gap for data collection in the Amazon. And we have this assumption that it might be this stronghold, this refuge safety net for a bunch of species, small cat species included. And it turned out that we didn't have a whole bunch of data sources from the Panthera or collaborator network to actually assess, these habitat variables that were specific to the Amazon. And so that was just that's a gap that I've come across in the last few years that I found surprising.
Right. And again, there could be a reflection of just how difficult it is to work in some of these habitats, right? You mentioned the Amazon's probably somewhere kind of very deep into that rainforest where it's inaccessible. It's very difficult to get those kind of cameras out. So yeah, no, absolutely. Any more thoughts? Yeah, Byron.
Sorry, but I was going to say one thing I'd bring out, I think, related to that, when you start talking about these refuge populations and refugial, context across the landscape, is what I think one major gap for small cats generally, and this is where they're easily detectable and not, is maybe better understanding population structure and connectivity. I know we use a lot of, with the camera trapping, you can get at those variables that can predict where you might, your habitat selection variables and things like that. And you can model how that structural habitat might connect populations. But what's lacking, I think, is actually going and saying, are they using it in the way that we're modeling it? And that is done, clearly, through genetics and like measuring gene flow and saying, all right, these are connected populations, or no, these are isolated and fragmented. And that adds another layer of conservation relevance to where you might target interventions and things, better understanding that. We're even at the point, I think, with small cats, and you all know this, it seems like every year the number of small cat species changes. Like, oh wait, we thought this was one species, now it's two. And so just that's like that broader phylogenetic scale of, phylogeography of like, we're still understanding species dynamics and taxonomic dynamics within small cats. So that's a whole other level of connectivity or isolation at the evolutionary scale that we're still resolving, because I think as a species, because it's so hard to study, that we're getting caught up to where, and the big cats and larger species in general that we're concerned with in a conservation context, like that data has already been sorted out. Those questions are already answered. And there's still some lingering, but we're still almost in the very early stages of resolving those questions for small cat species. And if we can't agree on taxonomic classification of a small cat, how are we going to assign, a red list status and those sorts of things to them in a way that's effective at the management scale planning?
Yeah, that's a really interesting take on knowledge gaps. Like you said, as we dive deeper into small cat conservation, we start, particularly on the genetic side of things, we start to identify more and more gaps. I think we actually get a lot better in closing some of the gaps, but as we do, it just kind of opens a lot more, and kind of creating a much more complex and kind of a diverse issue of how to kind of fill those gaps. But yeah, no, it's just that you mentioned genetics. I mean, so we have a group of species that is difficult to detect. They have low detections in the wild, very difficult to study. As a result, there are a lot of conservation gaps that's kind of impeding a lot of their kind of conservation plans. And so I guess one of the next tools is going into the final theme is what can we realistically do with existing tools, right? We have a growing toolkit available, camera traps, telemetry, genetics, eDNA, and increasingly integrated analytical approaches. But yet, as we're discussing, many small cat systems still remain somewhat data limited. And so Byron, was someone who has, extensive experience in camera trap and telemetry genetics, how can existing tools, monitoring tools, kind of either individually or combination, realistically help us address some of these challenges and support these conservation decisions?
Yeah, no, I mean, I think there's not any single one of these is like, if we could just do camera trapping better, then all we need to do is camera trap. So if we just did telemetry and everything, that's just not logistically or financially possible. And neither are genetics like some panacea of like this will resolve all of the problems if we just do this more. I think it really does require, and it's not a satisfactory answer, but it's a combination of all these tools. I mean, a really powerful sort of framework for that would be, for small cats in particular, is one where we're using camera traps to sort of define occupancy and relative abundance, trends. Telemetry can be used to define space use and habitat selection at those fine scales to really just know what these individuals need on that level. And then genetics can come in to find like who the animals actually are, like getting at distinctive populations or not, like how many animals are out there in a effective population size, so like in a way that's meaningful to population persistence, and whether those populations are separated in space, are they functionally connected or isolated? So I think I could combine them. It really is doing that's easier said than done. You're right. We're challenged ultimately by funding. Where's the money going to come to do all these things? But as you're developing, whether it's species-specific or regionally specific programs, I think the thinking in terms of how can I use multiple tools together to complement one another versus doing the one I'm comfortable with is going to ultimately reap better information and higher quality conservation action.
Yeah, absolutely. As I say, kind of, and as we know, small cats are just incredibly difficult to study. Very, very difficult to kind of pick them up on camera traps. As Andy's presentation the other day, kind of two detection of bay cats. So anything we can do to kind of layer on top of that to be able to kind of get those detections for us to be able to kind of perform that meaningful analysis. And so, Sara, from an analytical point of view, are there any challenges? Are there any issues kind of combining all those different kinds of data sets when it comes to, doing those kinds of analyses? Can you combine camera trap data with genetic data with telemetry data? Are there models out there that can handle that?
Yeah, I think, a model type that maybe hasn't been implemented much yet in the small cat world, but has been done, for large ungulates. It's being used more in large cat species as well, but could be of great benefit and great use is looking at ways to apply integrated population models. So in that case, you're looking at multiple demographic rates at once. You're using survival data, you're using recruitment data, you're using abundance estimates, all which can come from disparate data sources, and different methods for obtaining that data. And when you're using all of these data sources together, they kind of self-regulate, so if your abundance is decreasing over time, but you think your survival rate is very, very high, these rates are going to kind of self-regulate. And so using all of the data together at once allows you to get better estimates, more precise estimates for a variety of parameters of interest. And of course, there are challenges putting these sorts of data together. You want to make sure that you account for assumption violations of, each data type for each model. But I think integrating data is going to be a great benefit for small cat research and conservation.
Right, exactly. And so, Andy, someone who's done camera trapping and telemetry on the same species, any thoughts, any lessons there from you?
Well, just to respond to what Sarah was saying, it sounds fantastic, bringing together all those integrated approaches. But is that going to work for those really tricky species? We don't have survival data. If you go out there for 2 or 3 years and you come back with 10 photographs of an animal, and you might sit there and go, oh yeah, job done, we did great, and you pat yourself on the back, but what have you actually achieved? And this is the really tricky thing. I think for some of the small cats, we're going to really be able to progress over time and as funding becomes available, and we can bring in new methods, improve our monitoring approaches. But there's still going to be this cohort of small cats that remain to be stubbornly, unobtainably detectable. And so, the question remains, what do we do? We can throw more money at it. We can throw more cameras down, but there's only a finite number of resources. So do you just focus on those species that you can do something about, and at the same time you hope that there is an umbrella effect that it lends itself protection to those other ones that we just simply can't do anything about? Do we keep plugging away? I mean,
Yeah. I mean, it's a great point and a great question. I look to these are models that maybe have been developed more, they've been used more and developed more in the bird world, but are becoming more common in, a variety of guilds, modeling approaches, but working with community models where sometimes you can kind of share information between, if you have like kind of a cohort or a community of somewhat similar species. Some of those species are more commonly detected. Some of those species are super, super rare. And in the modeling approach, you can kind of share information and help still obtain a useful estimate for your parameters of interest for that really rare species. So that's, it's one idea that can be looked at is include multiple species in your model and use some of the information that you gain from the more common species and apply it to the rare species, just kind of from a modeling approach. I do think, as Byron said, there's some things that you're going to get from just specific methods. So like the telemetry data, you're going to get that kind of more micro habitat movement selection type data. You can get cause specific mortality. And I know there are challenges with, small cats and telemetry and GPS collars and even catching any individuals to put a GPS collar on. But I think you both point out that there are specific pieces of information that can only be obtained by certain methods, and it's going to be a challenge based on the tools that we have now.
Yeah. Yeah, it's a good point. I mean, for some of these really tricky species, and again, focusing on Borneo, the bay cat, the flat-headed cat, if we could, telemetry, of course, is a wonderful technique, but it's super expensive, tiny population sizes, and then our ability to extrapolate is limited. Nonetheless, when you do get those animals, they provide just unparalleled information that can then lend itself to perhaps improving approaches. Thus far, we haven't collared a flat-headed cat. Well, actually, no, there have been a couple, but just for a couple of weeks, I believe, in an oil palm plantation. Bay cat, never been collared. We've been trying. So, if anyone finds out how to catch these things, maybe that can help.
But I would maybe add real quick to that. I think, and again, I don't think this is the silver bullet that people have been hoping it would be, but eDNA is a rapidly developing technique that's going to help us, and especially in aquatic systems, which sort of mirror those really difficult places to like where to even put a camera. And these are arboreal species. Like, how do you set up a camera grid for animals that barely come down from trees? And so there's some methodological development there with the cameras, but it's also like, well, maybe that's not the method to use. What are other alternatives? And there's this rapidly developing methods within eDNA of paint rollers on trees, just simply rolling on trunks and branches to pick up DNA, of water sampling these semi-aquatic cats that are difficult to detect. And again, that's going to get you presence absence, which is better than nothing, and that's better than maybe what you're getting from cameras or from your occasional camera trap image of a tail flashing across, and like, well, it could be this species or that one. At least with the eDNA, if it's done right and you've invested in the databases, to reference databases, then you can get at maybe better certainty than you would anyways with the cameras. But those are still developing, and they require some level of investment that is expensive to get those up and running. Then I would add, like you say, like the occasional, it's difficult to do the coloring, but any given colored animal provides this wealth of information at that scale. I would say the same thing about a tissue or blood sample from an animal. Like maybe their cats are incredibly difficult to find and get a hold of to even get a good quality sample of, but genomics and the advances in genomic methods these days has gotten super cheap compared to what it used to be. And the genomic information from just a couple individuals can tell you, it's not going to tell you the entire species, demographic dynamics, but it can give you some clues about, if you have a handful collected from a couple of different places, some level of structure. Is there alarming levels of inbreeding? Is there, what's the level of genetic health of that animal that you can extrapolate to represent perhaps how that species is doing in that particular place? And again, not completely satisfactory and comprehensive, but can give you some insight into those valuable metrics on effective population and size and inbreeding coefficients and things like that. So I think, again, it'll always be with these rare species an unsatisfactory data set, but what we can eke out of little things by leveraging the right tools, I think, can really provide at least some informative bits of data to know how to move forward.
Yeah, absolutely. I think we're all kind of nodding our heads in agreement there. So fantastic. No, I'm really glad that kind of stimulated a nice discussion there. I'm a little bit conscious of time, and I can see in the Q&A that we've got a few questions. So Jolene, how are we doing? We're doing okay with time? We can open up for questions.
Sure.
Okay, great. So we have a question from Blair Hammond, if you don't mind unmuting yourself and asking your question to the panel.
Folks hear me okay?
Yeah, loud and clear.
Great, thank you. Super interesting conversation, and Byron spoke to some of my question, but I think the essence is camera traps have been amazing, but there are other techniques, and I do find over a career in wildlife biology that as a group we're pretty conservative in terms of how quickly we adopt new technology, and people still talk about radiotelemetry as high tech, and of course it's not anymore. And I'm wondering what the panel thinks about why we've been maybe a little slow to take up things like eDNA or hair trapping for DNA, when there have been a number of pretty good papers that have spoken to its cost effectiveness and its efficacy at sampling terrestrial mammals and also aquatic wildlife and other things as well. So what do you think is the barrier? Is it just that we hear DNA and we think, blimey, I'm not any good at that. I never was very good in my microbiology and so on, and I'll stick with what I know, and the camera trap is more accessible to more of us? Or what do you think might be going on with why there's slow adoption? I expected, honestly, to hear more about those techniques, especially for really almost impossible to survey. I like the invisible cats, where this might have some value, but I'm also ignorant of doing work on these species, so I'm curious what the panel thinks.
I mean, I'll jump in first. We were intrigued by these methods, and we've given it a go. I mean, we've been trying to answer these very basic questions for the last 20 years for some of these cats. So we have given it a go. We've tried to collect environmental DNA. We've put up, we've seen that there's effective ways to get hair and things like that. We tried so many ways, and it just hasn't worked for us. Some environments, and it might be us, but some environments are just very tricky for DNA. I mean, wet, permanently wet, hot. We've collected scats, and we've tried to run the scats, and we get very poor DNA results from them, such that we've only really started doing well with our clouded leopard research on genetics by getting blood, and that's from when we've actually caught the animals. But when we've used scats, we've just simply failed thus far to get sufficient results. So I think sometimes, and I'm sure there's a whole multitude of reasons, sometimes maybe some of these techniques are just are tricky in certain environments. That's certainly what we found.
Yeah, I mean, I can add to that I think that's just it. The places where the challenges are their greatest, it seems, are those where the environmental conditions are the most challenging for DNA in the environment. And Blair, you're speaking my language. I've spent a lot of time trying to talk people into adopting more diverse methodology that includes genetics. But again, Angie, you're exactly right that it comes up against these challenges. You try it once or twice or three times, and it's just not working. And then you just figure our limited budgets are better spent when what we're good at and have some marginal level of success versus investing further. But I think, again, we're at least there is a lot of investment in eDNA methodology, and environmental DNA in that sense is different than non-invasive sampling where you're collecting hair or scat or urine. So the eDNA, where it's like you're actually collecting unseen molecules of DNA that you go and take into a lab and figure out what they are later. And that's like through filtering water, that's through filtering soil. It's again, like the paint rollers on trees and collecting blood feeding invertebrates. There's another great way to collect. They're doing the sampling for you. You just have to go and sample them. So that as we get more inventive in how we look for DNA in these environments, we'll find more ways to detection. Again, we might not get at the type of data that allows us to say to track gene flow and understand there's more the more complex population genetic parameters, because that requires a higher quality template. But at least getting at presence absence, at least a way to compensate for where camera trapping is more challenging, and getting presence absence with eDNA. I think those will continue to move on, but they do require some level of a gamble of saying, let's try these out, let's trial these. It may not work out. We're going to move the process and methodology forward. And I think that's maybe where people are struggling now, Blair, is having limited funds to invest and not quite being able to take that leap in a developing methodology. And I think it's going to just take a while for that to catch up, as probably in reality industry moves those methods forward because they have all the money.
Yeah.
I would potentially just add briefly that there could also be a bit of a data processing and analysis barrier, like processing camera trap images, identifying species, identifying individuals, and implementing models like occupancy models. I feel like there's just so many R packages that make that very accessible. There's tons of workshops, tons of, online resources. So that's something that's very accessible and very approachable, I think, for a lot of researchers. And so, from a presence absence, obviously DNA could easily be plopped into occupancy models, but doing a mark recapture with individual identification, it becomes a little bit more tricky. And so potentially there's just a data processing and data analysis barrier too.
Yeah, no, great. Thank you. Great question there. And also, just a quick one from the telemetry side of things. That kind of technology has advanced so rapidly in many kind of aspects. You can tag bees, sea turtles, birds, but yet the collars that we put on our cats are still quite large and chunky. We try to maintain within the ethics to have kind of 3% body weight, but how do you even collar a flat-headed cat that's half the size of a house cat? I think there's one kind of type of collar that's out there, but again, it's very limited on the functionality of that particular kind of collars. So conscious of time, we'll kind of move on to the next question. We have a nice question from Nicolás Gálvez, if you don't mind unmuting your mic.
Hello, can you hear me?
Yep, loud and clear.
Hi, Nico Gálvez here from Chile. So in the red list assessments, we usually need to use the best available information. So where would you draw the line in using detection rates as a proxy to density, for example, in a particular assessment? Do you look at those particular studies where those estimates come from, their survey design, effort, spacing, etc., or just use the most conservative value, both, or other measures that you might think of where you draw the line, or you simply say, let's not use detection rates, for example? I don't know. Thank you.
Yeah. So in the assessments that I've personally been involved in, which is a number of Bornean felids and some other Southeast Asian felids, generally there's been pushback about using detection rates because of the problems associated with your inability to incorporate detection probability. So when comparing between areas across the landscape, it's very easy to criticize that rate and say, well, how meaningful is it? And because of that, we've tended to use other variables to make the assessment. So changes in assumed population size. So if we have a better understanding, we may have an understanding of the distribution of that or the habitat associations, and then we've used changes in that predicted distribution to really hone in on the species assessment. So generally, in my experience, yeah, we've just not used them, but I do think that they do have value on some occasions. So in the absence of anything else.
Yeah, no, exactly. I want to kind of follow on from that point. Like, you know, particularly with these red list assessments, we want to be as detailed as possible. Obviously, kind of population density, that is the gold standard. But for species where it's just not possible, you know, I do, yeah, like you said, it's better to report those if there's nothing else to do. And you know, Andy and myself are currently working on the flat-headed cat red list assessment, and we're kind of up against that similar sort of thing. You know, we can't report population densities, we do have detection rates. So we're kind of working out how best to kind of represent that section that allows us to be able to compare across sites and landscapes. I think we are running up a little bit up against time. Perhaps time for one more question. And we have a question from Victoria Barbosa, if you don't mind unmuting your mic and asking your question.
Yes. Hello. Thank you, Ming. Thank you for the panel, you all. I would like to know regarding methodology, like if we think in a scenario that we have like 1 million hectares but we don't have the resources to put the camera traps on grids that is 1 kilometer by 1 kilometer that was focused on small cats. But we could do like a 10 or 15 kilometers grid instead. Would be better to use this larger grid, like adjusting questions and models to respond the kind of researches, or reduce the area for a methodology that would be optimized for occupancy and density modeling, just focuses on the small cats, because we don't live in the ideal world. Like we don't have the resources to do that. But here in Brazil, we have this opportunity like with larger areas, but I'm really struggling to find some studies that uses larger areas for small cats, like to sample in a year. Most of times it's like 10 years that you divide the land, like in 10 years, 1 year you do this and do that. So if you don't have this time and if you don't have this resource, like in 1 year, what do you think that would be the best option? And thank you very much.
I would respond just briefly that if it really depends on your focus. So the last part of your question, I'm also reading it in the Q&A section. If you are focused on density modeling, I would definitely reduce your area and make your I guess I assumed it was camera traps. I don't know if you're using camera traps, but I would reduce your area and get more detections, more spatial recaptures of the individuals, and have to forego being able to survey the entire area. So if you're focused on density modeling, I would say definitely reduce the area and optimize for that focus.
Yeah, I mean, I think you would kind of need to think about the small cat biology here a little bit and how they move through the landscape. If you have your cameras far, spaced far too apart, you're going to increase the likelihood that cats are just going to walk in between these cameras. So, again, just echoing Sara to kind of use the smaller grid to increase your likelihood to be able to kind of get those detections. But, just thinking about your scenario off the top of my head, if you do want to go to that landscape, you could always think about, setting your small camera trap arrays with the appropriate camera trap spacing, but then moving that grid around the larger landscape, right? But of course, you talk about some of the resources, timing being one of them. That is a very time expenditure kind of method to deploy such a camera trap grid. But, yeah. Okay, so I think with that, we're out of time. And thank you so much, panelists, for all your insights and for all the kind of really thoughtful discussions that we've had around some of the thematic issues when it comes to measuring small cat recovery and how we study them in the wild. So thanks again for your time. And I hope that was a productive panel for the attendees and listeners. Thank you very much.
Thanks a lot.
Thanks so much.
Well, thank you to that amazing panel helping us to better design the ways, not just for now, but for the future as well. The critical information that helps guide the conservation and the recovery even of species. Now, just in some final reflections, sometimes I think folks think that we stand, if you will, in the shadow of attention that's given to 7 large cat species. Many of us work on large cats as well, but what I've seen here is that there's an incredible force for the conservation of small cats around the world. Indeed, I said, I think on the first day that you might have more in common with somebody in this symposium that lives across the world than a neighbor, because of the shared passion and commitment that you have for helping, these species. And I hope that in this symposium, you've not only been able to learn about the science and action, for small cats, but have been able to make some connections for the future, either collaboratively in terms of work or just in terms of supporting each other. Given the breadth of geography, of species, of actions, of looking at threats and all the different presentations, I'm thoroughly convinced that we've had, a huge proportion of the world's small cat champions, talking together over the last few days. And that's quite incredible when you think about it. We've also been dealing with overarching issues such as the sampling questions right now and analytical questions, but also looking at broader things like how do we grow global awareness, for these species? How do we engage with governments more effectively? And what is it actually that funders look for? And so I personally thank you for all your tremendous effort. I mean, in terms of putting the abstracts together in the first place, but I've been so impressed with the quality of the presentations and also with everybody keeping to time so well. That just shows what a high caliber we have. Of course, from a funding perspective, I also want to thank the Ayers Wildcat Conservation Fund, who's made all this possible. That's so crucial. Now, in our organizing team, you know what? We're all ecologists. We've never run a virtual symposium before. I want to give special credit to Jolene Broadfield, who was moderating today for just having done so much and corresponding probably with all of you to make this possible. Also to Angus and the Africa Massive team for working late into the night in South Africa, helping us so professionally and well. Thank you to you. Now, but because we've never done this before, and it's so funny because, people keep asking me, when's the next symposium? Never even thought about having another one. I don't know if it's even of interest. So one of the things is that we have a short survey, and when I say short, it really is short. So I'm actually going to give 5 minutes right now or 7 minutes to fill the survey that Angus is just putting in the chat, or has already, maybe. Angus, I can't remember now. And then I'll come back with some final words. But I'm giving you this time now because we have busy lives. People run away. We'll still, email some out as well. But I'd like your feedback. Angus, are we ready with that? There's just so many things in the chat. There we go. Small Cat Symposium feedback survey. Does everybody see it? Sort of 3 chats from the bottom. Yeah. So we're taking a break right now, but come back because there's a couple more things I want to say to you, if you don't mind. So 5 minutes right now to do a survey. Welcome back from our last break, last few words. Thank you all so much. It just feels amazing. And again, I mean, so many thanks to all of you. Just one thing I'd mentioned before from Panthera perspective as well is we're looking for additional partnerships, either for you to come partner with us on sites that we have. I gave some criteria in my first talk of what we're looking for, but really it's across regions, across species, across not just sites, but also in terms of general methodology, innovations, reviews. We're looking for it. So either you come join us where we are, or or if you'd like us to come join you and you know, bring our own resource. One of the things is you know, we're proud to be able to give some funding, and it was acknowledged throughout the symposium. We're happy about that, but we have even greater capacity in terms of working as a collaborator with you, and are most open to that. So contact us, you know, let's just see what's possible. And then overarching, you know, it's just 4 days of so many discoveries, exchanges, questions, connections. I'm almost, it's been such a big effort, but I'm almost sad now that we need to stop. But, you know, we need to stop sometime. And I hope that you all feel richer for the experience. Most of all, I want to say thank you to all of you joining, especially across all the time zones. I mean, here we've gone, you know, again, we've gone from Brazil to Indonesia. And thank you most of all for the small cats. I mean, the small cats that have no voice, and that need our help. And so thank you for what you do, and thank you for continuing to do it even when it's tough. And please support each other as much as is possible. It is worth it. You know, it's worth it for the cats. It's worth it for the ecosystems, but I think it's also worth it for the people around the world. I even think about children, you know, that treasure them. And even as I mentioned children, I think it is very much not just about now, it's about the future. It's about the future generations of small cats, but of people too. And so a huge thanks to all of you for all your time, your contributions, and for doing what you do, being who you are. Thank you, everybody, so much. It's been such a pleasure, and we will sign off. But keep doing what you do for the good of the world. Thank you so much. Goodbye.